What's up, AP Bio Penguins? So we are live for our CRAM session. Are you ready to rock and roll? Are you ready for the craziness? So, oh my God, there's now 264 of you. So, just quick reminder, I'm going to do my best to answer everyone's questions and do what we can. Miss McClinton is going to be in the chat
Switch to
helping you
Chrome
out
to
with
load your favorite.
everything that she can. And so we're going to rock and roll. We're going to do everything we can. I 100% know this is not going to be an hour. Like, it is going to be hopefully two. Hopefully less than two, but two is probably going to be like our minimum right now. It's 90 something slides, and I'm going to speed run at some points. Okay, I'm just trying to get through everything. I want to make sure that we can get what we can. And I'm a big firm believer on you do it right the first time. So like we've got a great resource for us. Okay. So welcome everybody. I'm so excited to have you all here. Okay. Since you're sitting in my live, you know you are a penguin, and that means you are dressed to success, which means that on May 4th, you are going to rock it. And I mean, like, you're gonna rock it, okay? So just quick couple rules, okay? Like, I don't normally do this, but I knew that there was gonna be quite a bit of people here today, so like, couple rules. Let's be respectful to each other, okay? I'm gonna be going through a lot of stuff. Let's try to keep the questions on topic. I know a lot of times you really want to get your question answered, so you'll keep writing the same question over and over. So let's not spam your question constantly. We're going to stay on topic. Whatever it is that we're currently talking about is what we're going to answer questions about. Let's be helpful. If you see a question in the chat that you know the answer to it, you can help each other and you can answer that question, okay? And then let's also be very thankful. I've got Miss McClinton in the chat. She's another AP Bio teacher, and she is phenomenal. And she's been in a lot of my lives, like in previous years. And she was in the Unit 8 one. So say hi, Miss McClinton, and thank her very much for being here to help us out. But let's get going with our session, okay? So we've had 8 days. What can you do? Okay. So I've got the Insta-Review from my Instagram stories. I've already gone through the whole review, but the whole thing is posted on my website. If you go under the Insta-Review, you'll find all the PowerPoints from all 8 topics. So you can find a topic that you're struggling with, and you can go through that PowerPoint and look through the questions and kind of quiz yourself if you wanted to. There's a 505-page review guide. Yeah, no, you can't do that 505 pages at this point, but you can do the multiple choice. You can do the free response that's in it. You can pick and choose the topics that you're weak in, and you can kind of fine tune those weaknesses with that review guide. I've got my FRQ Fridays recorded. They're all recorded all through from 2013 all the way to 2025. Every single year except for 2020 is there. So if you're working on FRQ and you're like, hey, how would be a better way to answer this? I don't understand how I could have gotten this answer. Like, those are all recorded for you, and they're all saved on, of course, YouTube for you. So just search the name, as well as I also have them just saved on my page. There's 120 weight ground games. So you're like, I just need to play a little game and review content. You can go through those games. They're always open. It's just a straight link to them. And then, of course, I've got the review videos. I've got a review video for every single unit. Has been remade this year in 2026 for that new CED. Plus, of course, I've got the PowerPoints and everything there. And speaking of those PowerPoints, if for some reason I'm covering up some of the content in that top corner, I do my best not to, but sometimes it happens. The whole PowerPoint for today is already posted on there. If you go under 2526 review, you scroll all the way down to the bottom, you will see the PowerPoint, as well as that's where the link is for this recording later, okay? So other helpful resources, I usually do this at the end, but let's do it at the beginning. If your teacher has not opened up AP Classroom, say, hey, can you please open up those progress performance checks? This will give you a little bit of practice with AP style questions. Also, there are, going from 2023 also through 2025, FRQs are all available. It would be a good idea to maybe pick one of those years, set a timer for 90 minutes, and then just practice through a set of FRQs, giving yourself that timeline. And I hear my children talking about background, so just, if you hear random chatters, I have kids, it happens, I'm sorry. Also, if you're like one of those who likes to run or walk, podcast, The Absolute Recap has an amazing podcast going through every single topic that's in the CED. And then, of course, we've got Review Book. My favorite one is the Barron's. At this point, we're 8 days away, so I would probably go see if a friend had it, an upperclassman had a book, or maybe talk to the library. There's probably some copies somewhere. Maybe your teacher has one you can borrow. I wouldn't go on Amazon right now and try to buy a review book, but if you've got one, use any books you got to give you some extra practice, okay? So the plan for today, we're going to talk exam format. We're going to talk some tips and tricks, okay? We're going to talk experimental design and then graphing, okay? I know that this is some of those topics that we don't normally use, okay? So we're trying, not really that we don't use them. We normally don't talk about them. So I want to go ahead and hit those first off and get them really going. As well as I've got a rapid review. We're going to talk very quickly going through unit 1 through 4, okay? If I go too fast or you're like, I really need to hear a little bit more information, you are welcome to go to one of those longer videos that I've gone through that whole topic, okay? We got our practice questions. I've gone through the CED and I pulled up every single question. I aligned them to which of the units. Well, actually the CED aligned them, but I aligned those questions to have them ready for us. So we'll do all those multiple choice practices. And then we'll do rapid review going through 5 through 8. And then we'll do practice questions again, 5 through 8. And then if we have time for questions, but I don't think we're gonna have time for questions, but we're gonna do our best, okay? So without further ado, what is the format? Okay, you are going to be taking a hybrid exam. Hybrid exam meaning that it is going to be on the computer for your questions and your answers will be in the computer for multiple choice and you'll be handwriting your free responses, okay? So Blue Book is where you're going to be taking that. If you have not downloaded Blue Book yet, you want to go ahead and download Blue Book so you can get a feel for it. What does it look like? How does it feel to work through that platform, okay? So as I said, the entire multiple choice section, is going to be on your Blue Book app. While your free response, you'll have the questions in there, and then you'll respond in a actual like physical book. Okay. There are 60 multiple choice questions. You'll have 90 minutes for section 1. There's 2 long and 4 short FRQs in our section 2. And as I said, you're going to be writing in a physical book. Your second question will be graphing. And yes, we have 8 days to the AP exam. May the 4th be in our favor. So that's why I'm wearing a mandatory shirt today, is just to support us for our May 4th test that we've got coming. So this is what the application looks like. When you go in right now, you should be able to go through the preview, and so you'll click on that test preview and you'll select what we're taking. We're taking AP Bio exam, so you go through there. It kind of shows you what the directions look like that day, so you can kind of look through what are the directions going to look like. You can go ahead and read the directions now, so you're not going to have to read them on day of and lose that valuable time, okay? You notice there's also a timer that's at the top of the screen, and so that's going to be there, and you can hide it if you need to during the exam, but you don't have to worry about bringing a watch. You don't have to worry about that there is a clock at the front of the room that you can't see. You have a clock in front of you, and your exam is timed based on you, so when you are able to get into the platform, you'll be able to have your 90 minutes. Okay, so then why is it not cooperating? Okay, so within that app, you also have a Desmos calculator. I recommend bringing your own calculator, but you do have a calculator in there if you need one, as well as I have a copy of the formula sheet here. After last year's loveliness, they should be giving you a physical copy of the formula sheet, but this is a backup in case you need it for some reason, okay? As well as there's also some stuff under that More tab. There's some shortcuts. There's a line reader, which I think might be helpful if we're looking at like a graph. You can use the line reader and find like a specific point that you're looking for, as well as you should be able to zoom in and zoom out. That was one of the quick keys that I saw that might be helpful. So Control plus and Control minus will zoom in and zoom out for you. So that will be a helpful tool. So in terms of our multiple choice, okay, as I said, there are 60 multiple choice questions. You have 90 minutes, okay? You have about 1.5 minutes per question. So every 10 questions, check your clock. You should have had 15 minutes elapse. And so I'm assuming that the clock is counting down, not counting up. So after 10 questions, you should be at 75, 60, 45, 30, 15, and of course you'd be at 0, okay? If you are running behind schedule, you just got to speed up a little bit. If you're okay and your timing is okay, then just keep on doing what you're doing, okay? There are 2 different types of questions we'll have. There are individual questions, so it's just like a straight question with an answer. There'll just be a short little prompt. And then you're going to have set questions. Set questions will have a longer prompt. It's usually going to have some type of diagram or graph that you're going to be looking at. And then there'll be about 4 to 5 questions that go with those, that graph or that table or that long prompt, okay? So in terms of multiple choice weightings, okay, so you can see the weightings. I kind of set them at the beginning of each of my lives that I did for each of the unit reviews. You can kind of see that the heavy hitting units would of course be our 3, our 6, and our 7, okay? In terms of the 2025 exam, these were the numbers that we actually had. So they did have a lot more that were hitting in unit 4. So 4 does tend to be a little bit more because of that self-communication, but 10 is really, unit 7 is really where we see a lot of those questions. In terms of science practices, we do have a lot of just straight content explanations, as well as we have a lot of argumentation. So applying that information in a higher sense, not just straight, do you know the biology? There's a lot of applicational type questions that go with that. And so in terms of our multiple choice, we want to highlight any of the important words. You have a highlighting tool within your Blue Book app. So highlight anything that's important as you read through the prompt. If there's anything you want to jot down, you have, I think, 4 or 5 pieces of scratch paper, so you can write down that scratch paper. Cover up any of your answer choices. Like, so a lot of times, like, you can develop the answer on your own. Like, if you go through the question, you're like, oh, I know this is XYZ. Go ahead and look at the answer choices and see if that's there. Okay. Oftentimes there'll be answer choices that distract you. So if you already know the answer, you're looking for that one. So that kind of helps to alleviate a little of that distraction. You don't need a title for your graphs. Use the figures or the diagrams to help you answer the questions. Okay, so a lot of times you can answer like the question by literally looking at a diagram or looking at a picture. Like, so like, let those diagrams help you. Don't freak out if you know, if there's a word you don't know. They're going to throw chemical names or diseases or different things, or hormones and, you know, signaling molecules, and you don't know those. Just pretend like they're an X or a Y and be like, chemical X did this. Cool. Like, you don't need to know the name of the chemical. Just kind of work with it. Okay. Annotate your diagram. So you're not as able to annotate, but there are little like ways you can like type into the screen. So if you want to like make quick notes yourself, or just write it down on your scratch paper. Okay. Show your work. Oftentimes as you're rushing through things and you're just typing things into your calculator, you make mistakes. So if you write it out on your scratch paper, you're less likely to make a mistake when you go to type it into your calculator to do your math. Okay. These multiple choice questions, just 1 point per one. So you really gotta make sure you get as many points as possible. If you see a question, you're like, oh my God, it's asking me to do an entire chi-square question. Go ahead and make a guess and mark it for review and come back when you have more time to go through a full chi-square question. And then of course, watch your time. As we said, that's backwards. Anyways, every 15 minutes, you should go through 10 questions. Okay. So in terms of our second part, our FRQ, okay, there are 6 questions, 2 long, 4 short. Okay. So in terms of timing, give yourself about a 5-minute reading period, go through all the questions and read through them, then spend about 20 minutes on your long FRQ, and then do 2 of your short FRQs, spend about 8 minutes, and then do another long, and then the other 2 shorts. Okay, so you can renumber them. You can do them in any order you want. Okay, if you do 20 minutes and 8 minutes for your longs and your shorts, that gives you about 10 to 15 minutes at the end that you can go back to any of the questions that you need to sit there and think about, or you want to add a little bit more information to them, or kind of revisit any of them. And I'm sorry if I have to take a couple sips of water every once in a while. So in terms of the worth, I've been wrong in every live, and I'm so sorry about that. Apparently, with the new CED, they now decided that every single one of the longs are now 9 points. So if you look at the CED, it says that our question 1 and 2 are 9 points, and then questions 3 through 6 are all going to be 4 points. Okay. So I did an activity with my students recently, and so my advice would be that you want to do questions 1, 3, and 6 first. So those are the questions that have a lot of identify the null hypothesis. What is a control? What is an independent variable? That's where a lot of those questions are. And so those are questions you can get without knowing the biology. Those are what I call our easy points, our low-hanging fruit. Go ahead and get those points and move on. Okay. So these were the directions that were on the 2025 packet that's on AP Classroom. AP Central. So I'm assuming this is what your directions are going to look like, so you can kind of, read through them in advance again. That way you won't have to worry about reading them day of, because you've already got access to them. Okay. It does talk about labeling the different parts. You can have a calculator, and they also talk about that you can use pencil. Okay. So this is what the booklet looks like. So you're going to open up your book, and it's just going to say begin question 1, and you'll write your response. It also says do not skip lines. What that means is don't skip every single line. Like, you can write a paragraph and then skip a line, write a paragraph, skip a line. So don't freak out when you see this do not skip lines. You're like, oh, I've already skipped a line. That's not going to happen. You're perfectly fine. You will be they will grade it no matter what. They just don't want you skipping lines, because then you run out of paper, and they want to make sure the whole response will fit on the pages that they've allotted for you. Okay. So then you go on. Here's our question 2, and it says again, use only one color. So your scans for your graphs are black and white. The readers cannot see different colors. So if you're using a legend, you're like, blue is going to be this sample and black is this other sample. The readers can't tell the difference between your colors. So you got to find some other way to differentiate your different parts in your legend. One of them shaded, one of them's unshaded. That's perfectly fine. And then again, we would go on to question 3, and it would keep doing this all the way through question 6. Now, here's the thing. Sometimes you get rocking and rolling, you're going through it, and then all of a sudden you flip the page and it says begin question 3. And you're like, but I already wrote question 3. That was on the page for question 2. And you freak out. Y'all, do not freak out. If you accidentally write your response on the wrong page, it happens. You are in the moment, you're rushing through this exam, you're doing your best to try to get as much as you can onto that paper, showing off all that amazing biology that you have, okay? So what I need you to do is just go to the question that you're supposed to be writing on. So in this case, since my example was about question 3, go to question 3 and say, please see question 2 for response. Your reader is going to go full book no matter what. That is our part of our protocol, is to go full book if your answer is not where it's supposed to be. Okay, but it helps your reader ensure that they find it. Okay, they're going to look for it, they're going to read through it, they're going to try to find it. But it does kind of help them out a little bit, and you know you want to help your readers out. So different tips for our FRQs. As we said, read the question. I don't know if you heard me. I said read the question. Y'all know so much biology that you want to show off all that biology, and then somewhere along the line you start answering a question that's not actually there. So label your responses. So part A, B, C, D, and you can answer them in any order. So if you know part C, C, answer it. Oh, I know A, answer it. Then I know D, oh, D, answer. Oh, then I don't know which one I didn't say. B, answer it. So it's perfectly fine to write them out of order. Just label your sections. It makes it easier for the reader, okay? And don't restate the question. We know what the question is. We don't need you to tell us the question, okay? Now, if it's like an identify one, and it's like identify the chemical, the da-da-da-da-da, just write the chemical is blank. Yes, I know on identifies you could just write the word, but it does ensure that you're going to have as much as, like, you're going to ensure you get the point by telling them enough. Laser, it's going to be as long as it is. I'm going to do my best to get through it in as fast as I can, but I'm like 90 something slides. So if it asks you for like 2 of something, you want to say first da-da-da, second da-da-da. So it tells the reader what your 2 are, because sometimes as you're writing a response, you might accidentally put 2 of your answers there. So it does allow you to like kind of differentiate which are your 2 that you're trying to label. And beware of contradictions, okay? So if you say the population increased, and then you go through your justification, you explain why the population increased, and at the very end you go, the population decreased. You just contradicted yourself, and now that's gotten you to not have your point, okay? Use diagrams. A lot of the times the answers can be in your diagrams. Define your terms. Don't just name drop. Don't just be like, oh, it was natural selection. Well, describe how, describe why, justify, give me some meat. Show me you know what natural selection means, okay? Make sure you have the correct type of graph, that you give it a scale, that you give labels, okay? So write clearly and legibly. We're going to do everything in our power to read your response, but the more clearly it is, more legibly it is, the better it's going to be, okay? If something is not to be scored, just go ahead and write a line through it, just cross it up, okay? And then you're going to use a pen for the entire FRQ, all portions of your FRQ, except for your graph. If you get to the graph, well, when you get to the graph, you're going to do graph it in pencil. We know you're going to make a mistake on the graph. It happens. We're humans. We make mistakes, and you're rushing through this exam, okay? So use pencil. That way you can erase it, okay? You want to not use an erasable pen, okay? We need you to have a real good Bic pen. Like, this is a perfect pen for the exam. Do not get those fountain pens, those fancy, Sharpie pens. Dude, a Bic pen is perfectly fine for you, okay? So you want something that's not going to bleed. You want something that's not going to smudge, because these things are going to, of course, you're writing quickly, and then your hand's potentially going to go through it. Also, we want to make it so that it doesn't bleed to the other side, okay? So watch your time. As we said, 5-minute reading period. Renumber it, 20 minutes for all your longs, 8 minutes for all your shorts. And then this is what the front of the blue book looks like. I'm sorry, the reference, your book. The book you're writing in, okay? It does say right there, use a pencil. So if you accidentally are writing your FRQ, like you forget to put the pencil down on question 2 and you're still writing in pencil, it's okay. Switch out the pencil for the pen, because the fact that they can grade it in pencil. It's perfectly fine. We just prefer the pen because the scans are a little bit darker. You can't use, or you shouldn't use, an erasable pen because the heat is what's going to get rid of it. So as it goes in the the shipping to get there, it sits in the hot warehouse. There's a chance that maybe your ink could erase. I don't know if it will or won't. That's just a fear that we all have, irrational fears, So experimental design, let's work through this. The independent variable. The independent variable is what the researcher is modifying, okay? So in the question, in the prompt, they're going to tell you the researcher changed this. The researcher did that. That is your independent variable. Okay. Then they're going to say the researcher measured this, or you'll have a data table with the measurements in it. No, you can't use whiteout. You're going to have it's going to be in the prompt, okay? So what is the researcher measuring? That is your dependent variable. Your negative control is the experimental group that does not have the dependent variable. So we took the dependent variable out because that's going to allow us to see what is that dependent variable doing, okay? A positive control is going to be one that has a known result, okay? I know that a plant needs light to grow, right? So if I give it white light, it's going to grow. And then I can compare that to a red light and a green light and a blue light because of the fact that that's going to show me that it's going to grow. I can't just put a plant in the dark because, well, plants won't grow in the dark. So you need to have just light, and that would be my positive control, the white light. Okay. And then the null hypothesis, I always use it in the same format. Independent variable has no effect on dependent variable. Okay. So whatever it is that is being changed had no effect on the actual outcome of it. Okay. And so I went through some of the exam. Let's see if we can pull out some of this stuff, okay? So the researchers studying two different types of protein transport identified that the ER membrane protein SR is necessary for transport during translation, while the ER membrane protein SEC62 is necessary for transport after translation. To investigate which transport mechanism is used for different proteins, researchers first created small interfering RNAs that reduce the expression of either SR or SEC62. They then treated groups of cells with either SR siRNA or SEC62 siRNA and determined the relative amount of SR and SEC62 protein in each group of the cells comparing with cells treated with the control siRNA. Go ahead and pick a color, okay? And so what you can do is you can highlight in that prompt the color. So you can always pick yellow or blue or whatever colors you want for the different variables. And if you use those colors, then you can quickly, when they ask you the question, you can go back and find the answer very quickly, okay? So you also see it on the graphs. You can see on our x-axis, we've got those different siRNAs that were added. And then you can see that we're measuring that relative protein content, okay, on our y-axis. So identify the dependent variable in the experiment shown in figure 1. So here's figure 1. Identify the dependent variable. Oh, well, the dependent variable is going to be the siRNA that's been added to, I'm sorry, dependent variable. Dependent variable is going to be our y-axis. I'm so sorry, I'm Russian. So it's our relative amount of our protein or our protein content. Okay, so looking at that y-axis, we can see that that was the dependent variable. That was what they were measuring in the experiment. Okay, justify why the researchers included the control of measuring the relative amounts of both SEC62 and SR proteins in the cell that were treated with SEC62 siRNA only. Okay, so why are we even looking at siRNA? Well, siRNA is going to reduce the expression of the SR or the SEC. And so by us putting it in that one, we're going to be able to determine whether that siRNA actually did reduce the content or not. We're going to be able to have that control to determine, yes, what happened? We did see a decrease, and it should have decreased. So this one should have decreased, and this one should have decreased because we were restricting and we were reducing the expression of it because I used the SEC62 and the siRNA that was for SEC62. So there should have been a decrease here, and there should have been a decrease there. And so it's ensuring, did the experiment do what it should have been doing? Yeah, I said the wrong thing. I'm so sorry. No, I've seen questions where the dependent variable is not always on the y-axis. The old CED, and I don't remember if it's still in there. I didn't look that deeply today in the CED, but I think that the FRQ question that's in there, no, it's still in there. They rotated that one. It's about some type of bug, and they like the bars come out from the side, and so it's a little bit different. Yeah, I talk fast, especially in this live stream, and we're at 23 minutes. So researchers then measured the amount of each of the 3 different proteins that was transported to the ER in cells treated with the SEC62 siRNA and siRNA. The researcher calculated the percent transport relative to the cells treated with the control siRNA. Okay, so again, we're looking at our x-axis to say, oh, well, they added different siRNAs. We're again still measuring the relative amount of protein that was transported to the ER, so that would be our dependent. So identify the independent variable in the researcher's second experiment. Now we're on the x-axis. So it's going to be looking at the siRNA, or that type of siRNA that's been added to the cells. Now here's the thing. That's not the only independent variable we have. Look here. We also tested 3 different proteins. And so you could have also said the type of protein whose amount was measured. Okay, so as you're looking at 3 different proteins, you're able to then compare those 3. So now, based on figure 2, identify the proteins that when treated with SEC62 showed an increase in the percent transport to the ER compared with the control. Okay, so again, we're looking at SEC62. Okay, and so it's comparing it to the control already. Here's that 100%. So we're wanting to see which one is going to show an increase in the percent. So which one do I see that is over it, right? So here we see that one is under. So it's not protein 1. Here I've got that it is over that line, so there's protein 2 and it is over. And here I have it over the line again, so protein 3 is also an option. So proteins 2 and 3. You could have said just 2, you could have said just 3. And the reason why I'm going through these is just so you can kind of see how you can get points when you don't actually know biology, right? We know biology, we're good at biology, don't get me wrong, but we want to get these easy points. So that's why I'm going through these kind of questions first, just so we can go ahead and see these strategies and how to work through these, okay? So then, oh gosh, there's math. Miss Jones, you didn't tell me there was going to be math in this. Sorry, y'all. So sometimes the math that we have to do isn't something that's on the formula sheet. And so I want you not to freak out when you get to those kind of questions. So protein 1 is encoded by 234 nucleotides, while protein 2 is encoded by 495 nucleotides. Assuming all nucleotides for both proteins encode amino acids, calculate the difference in the number of amino acids between the two proteins. What? I have to figure out amino acids. Well, how many nucleotides are there in an amino acid? Oh, there's 3, right? So there's 3 nucleotides, right? One codon corresponds to 1 amino acid. So I have a 3 to 1, right? So there's 3 nucleotides for every 1 amino acid. So if I need to convert this into amino acids and find the difference between them, then I'm just going to first divide them all by 3, and then I'm going to subtract, okay? Y'all, let's not have beef in our chat. There's 633 people in here, and I'm assuming y'all want to learn this biology. So let's just be good. Please and thank you. So protein 1, 234 nucleotides, okay? And I'm just going to divide by 3. If you've never seen this, this is a dimensional analysis. You're going to multiply across the top and divide across the bottom. So 234 divided by 3 gives us 78. We do the same thing for protein 2. So we do 495 divided by 3 gives us 165. It said we're trying to calculate the difference. Difference is always going to be subtraction. So 165 minus 78 is going to give me 87 amino acids. So the answer to this was 87 amino acids, okay? So then let's look at question 3. Scientists conducted an experiment to determine whether they control the abundance of buffalo grass population. The scientists identified several native grass species that, when grown with buffalo grass, might reduce the abundance of buffalo grass. They grew buffalo grass in the presence of several different native grass species in greenhouses in either the non-drought watered every 3 days or drought watered every 9 days conditions. After 12 weeks, they measured the height and the dry weight of the buffalo grass in each of the treatments. So we need to identify a control, right? So the control was going to be where we're going to look at that it doesn't have that independent, it doesn't have the variable to it, doesn't have the independent variable, right? And so if we're looking at native grass and it's either drought, non-drought, we're looking at it in the presence of that native species. So we want where it's only the buffalo grass, right? We want it to be where there's none of those native species there, because I want to see what will the buffalo grass do when it's by itself, so I can compare a lone buffalo grass to buffalo grass with the native species, okay? Then I need to state the null hypothesis. Again, the null hypothesis, the independent variable has no effect on the dependent variable, okay? So there will be no difference in the effect or the abundance of the dry weight, height, size, right? So here is our dependent variable, right? There's no difference in our dependent variable when it's grown with the native plants, right? So our independent variable is growing in the native plants, okay? We also have the independent variable looking at that drought and non-drought conditions, okay? So again, there's no difference in our dependent variable, the abundance, the dry weight, the height, the size of buffalo grass, when it's grown in the drought versus the non-drought conditions. Again, we're looking at the independent variable has no effect on the dependent variable. So here we're looking at question number 1 from 2022, okay? It has to do with that there's this cholera, okay? So the scientists are going to study the effects of the cholera on 4 different samples. They contain the G protein related signaling transduction pathways. GTP was added to samples 2 and 4. Cholera was added to samples 3 and 4 only. They then measured the amount of cyclic AMP produced by the adenylyl cyclase. And if you don't know what these words mean, it's perfectly fine. On this one, they actually gave you a diagram, so it's perfectly fine. Anyways, so I need to identify the independent variable. Independent variable is what did the researcher change? The researcher changed the amount, like whether there was GTP or cholera in the presence, right? So the presence or absence. If you look here in this chart, I've got a plus for presence and a minus for absence. So is the presence or absence of cholera toxin, the presence or absence of the GTP, okay? So it's the presence or absence of that would be our independent variable, okay? So then our next question was identify the negative control. Negative control is the one that does not have the treatment in it, right? So if you look here, what is the one that is absent from everything? Oh, look, we're missing the GTP and the cholera, so it doesn't have that dependent variable. And so you're going to see that I've got sample 1 would be my control, okay? Now, there are other options. If I'm looking at that the independent variable was just the cholera toxin, then I could talk about how 1 and 2 both are missing that toxin. Or if I'm talking about the absence or presence of the GTP, then I could be looking at 1 and 3 being my control. I also could look at just 2 alone. So there's a lot of different options you could have put here, whether it was sample 1, 1 and 3, sample 2, or 1 and 3 together. And I don't know why I went with the Roman numeral there. I'm sorry, y'all. So Based on the data, describe the effects of cholera toxin on the synthesis of cyclic AMP. Again, how can we use the data and just answer the question? Okay, we don't need to know what cyclic AMP does. We don't need to know what illness cyclase does. All we're doing is using the data and trying to find the information from it, right? So describe the effects of cholera toxin on the synthesis of cyclic AMP. So if I look here, this one does not have cholera, and this one does have cholera, right? So I can see what the effect of cholera was. Oh, look, 0.5, 0.5. The cholera has no effect on the cyclic AMP production. But here's the thing. It's both going to be in the absence of GTP. When there was no GTP present, then there was no difference. But if I look at 2 and 4, now when the GTP is present, I go from 10 to 127. So when GTP is present, cholera does have an effect, and it's a positive effect. It's going to be increasing our production of cyclic AMP. Miss Jones, I thought you were done giving us math. Sorry, y'all. We have more math. So percent change is another formula that's not on our formula sheet. Okay. So you need to know the percent change, and you did this when you did your osmosis diffusion lab. So you should know this formula, and it has shown up on the FRQ section multiple times. And so students oftentimes don't know how to do this formula. So we want to make sure that we know this formula. Okay. So our percent change is going to be our final minus our initial over our initial, and then we're multiplied by 100 to get it into percent, okay? So what we're going to do here is we're looking at that originally we were at 10, and then we're at 127 because we're looking at samples 4 and 2, and it says that we're going to see the percent change in cyclic AMP production in the presence of cholera. So my final is going to be 127 because it has cholera, and my initial is going to be the 10 because it doesn't have cholera, okay? So we're going to do 127 minus 10 over 10. 127 minus 10 is 117. And then you divide by 10, that's 11.7 multiplied by 100. I think I just said the wrong number. I'm so sorry. And that gives us 1,170%. That's a very large number. And sometimes students get freaked out. So just double check your math, make sure it's okay. But don't freak out if your numbers are crazy. The other time that we saw this, the number was like 3,000 and something. And then it was like negative 90 something. So like the numbers do look a little weird sometimes. Just trust yourself. Like you are geniuses. You are smart. You are paying with just success. Like you got this. Okay. So in terms of graphing, because I know you all want to love graphing, right? So straight out of CED on pages 184 through 188, there are all of the different graphs that you could potentially see on the exam, okay? And so here is the standard, okay? It says construct a graph to represent the data. And so you need to be able to construct any of these graphs, okay? The type of graph is appropriate for the data. You have to be able to label your axes, have appropriate units and legend. You have to be able to scale it, properly plot it, and then have error bars when it's appropriate. I don't have chi-square built in. I was going to put it in there, but I already had 90 something slides. And then a trend line when it's appropriate. So here, this is a bar graph. How do I know that it's going to be a bar graph? Because of the fact that I'm looking at categorical data. It was sunny and it was shady, okay? Those two data sets, that's categorical. There's no like linear scale to it, okay? And it shows comparisons against that categorical data, and it compares two variables. Versus with a histogram, you're actually going to see some type of spread of your data. There's some central tendencies to it, and it's looking at one variable. We're looking at the diameter measured here, okay? And so you had these little groupings, and so you were looking at kind of the central tendency to it, okay? This is a line graph. If you have a line graph, I need you to actually connect your dots and make a line, okay? So it's used to track changes over time. There's a series of our continuous data points. We have a linear scale. We're going from 0 to 20 months in a linear system, okay? You will not be asked to calculate the SEM. That has come straight from people from College Board. You will never be asked to find standard deviation or the SEM. Now, they may give you one SEM. You have to multiply by 2 to get to 2 SEM. SEM is the standard error of the mean. It's how you get your error bars. So in case of this one, it's just showing you the stretch of the data. The guppies at time 0 had a large variance in their genetic information. So they had a lot of different, like some of them had a lot of spots and some of them didn't have too many. And then we saw that over time, they kind of decrease that variance, and so you ended up with like a smaller SEM. You don't need to have a title. It's perfectly fine to not. So here is a log scale graph. So you're going to use a non-linear scale. So if you see on the bottom, we go from 10 to 100 to 1,000 to 10,000. It's so that you can get everything graphed on that data, on that graph, be able to visibly see it all. Okay. You have a dual Y, and so you could be looking at like predator population on one and the prey population on the other. That would be a dual Y. And keep in mind that your bar graphs can also be a dual Y graph, okay? So it doesn't have to be a line. It was just this example wasn't a line graph. We also have scatter plots, okay? So a scatter plot are just going to data, like you'll plot each of the data sets, even though each of the points, and then you'll draw a trend line. Those of you freaked out because you don't get to have a ruler, y'all, you can use the side of your scratch paper as a straight edge. You can use the side of your book as a straight edge. You could use your other pen as a straight edge. So there's a lot of things near you. If you're freaked out about not being able to make straight lines, you have the tools in front of you to make lines straight. Okay, and then we have a box and whisker graph. Okay, some of you have never seen box and whiskers before. So the box and whisker is actually showing us like all of the data. Okay, so this is the first top 25% of your data. Then the box all the way to the line is the next 25% of your data. The line to the bottom of your box is the next 25%, and then the bottom of the box to the end of the line is the other 25%. This is not showing you your error bars. This is actually showing you your data and the spread of your data. The line in the middle is showing you your median. Okay. There was a question number 6 on the 2024. It was recently where you actually had to analyze a box and whisker. And then we have pie charts. Pie charts are actually going to compare different parts of a whole. So we can say like the amount of individuals, number of cells that are in mitosis, like in all the phases of mitosis. You can do it with these different treatments. And so it's a whole that you're breaking up into these little percentages. Okay. So in terms of what are the graders looking at, this is straight out of the CED. They tell us that you have the right graph, right? That you've labeled it appropriately. Your x-axis has your independent variable. Your y-axis has your dependent variable. Okay. You've got a scaled interval that's uniform, okay? Pick a number that is logical, okay? You cannot count by 0.67. Like, I know you can count by 0.67, but it's a pain in the butt, okay? So count by something logical. Count by ones, count by fives, count by twos, count by tens, count by hundreds. Count by something that's logical for the data that you have. That is going to stretch as much of your graph as possible. You are also able to break your axis, okay? You do not have to start at 0, 0. If you don't want to start at 0, 0, you want to start at 40. Just write 40 at the very bottom and then just go from there. Make it so you use as much of that graph as possible, okay? But you don't actually you don't squish your data. You want to make it so that they can actually see your data points. If they can't differentiate your data points, they can't appropriately determine, do you know how to graph, okay? And then everything is identifiable, and then the error bars are on there, okay? So here we have the average general orientation of the activity. So what I need you to do is think to yourself, what kind of graph would this be? Based on the fact that I have male moss injected with saline, I've got male moss injected with siRNA, I've got general activity and oriented activity. I'm going to give you 3, 2, 1. This is a bar graph, okay? So you would have the activity of the moss on your y-axis, and then the x-axis was going to be either the saline, so your control solution, or your siRNA. But here's the thing, you could have also done it a different way. You could have put your general and your oriented on that x-axis, so either one is fine. Notice that they did shade in their bars, okay? So they've got one that's completely shaded and one that's just got these little hash marks in it, and they made it a legend that shows that, okay? Now, as you're looking here, you can clearly see the bottom of that error bar, okay? So it's perfectly fine to cover up that bottom, but make sure that it is able to be seen. You can visibly see what that bottom is, that you've not like darkly shaded your bar. Make it very light so you can still see the error bars, so they can appropriately grade your error bar, okay? Sorry, the shaded bars do not distract from the bottom of that error bar. So as I said, you're not taking away from that, okay? So based on the data in table 1, determined the type of activity that was affected by inhibiting the expression of the whatever DOP thing, receptor, okay? So here I'm looking at that I've inhibited the expression of the DOP, okay? So determine the type of activity that was affected, right? So that's going to, of course, be my oriented activity. As you see, it has 25, but the control was, of course, 20, okay? So we see that it is going to be decreased in that, okay? I'm sorry, I compared the wrong thing. 60 and 25, you see that one was decreased versus here, 95 and 90. They're basically the same, plus their error bars overlap. So you can see that it was the oriented activity that the error bars did not overlap. So that shows you that it's the oriented one that's affected, okay? So based on the data in the table, identify the treatment group in which the oriented activity was greater than 50%. A student of mine asked this the other day and was having trouble with it. Y'all, they just wanted you to look at oriented activity and tell me which of these numbers is greater than 50%. I'll give you a second. Yeah. It was the saline one. So control, saline treated group, the male moss were injected with saline, the control. So of course we're seeing that it's the one that is greater than 50, which would be our 60, okay? So here we see the rate of the oxygen consumption and the ATP synthesis. We're looking at different temperatures. We got 20, 25, and 30. So where in that, what kind of graph would I make on this? I'm going to give you 3, 2, 1. This is a bar graph as well. So here you can kind of see they deduct one that was unshaded, being our 20 degrees. They made one with just hash marks in it to be our 25, and then they shaded in the other one to be our 30. Okay, and they did theirs based on the metabolic markers. So the oxygen consumption was one, and the ATP synthesis was the other. And then over here, this individual did it by temperatures. They did 20, 25, and 30, and they shaded based on whether it was oxygen or it was ATP. Okay, so you could have done either one. So I blew up the graph a little bit and used one of the sample ones instead. So based on the data, determine the temperature in degrees Celsius at which the rate of oxygen consumption is different from the rate of oxygen consumption at 25 degrees. So here we see, right, our 25 degrees is right here, right? So we're trying to see which of them with the oxygen consumption, so that's the unshaded one, right? Which of them is the error bars not overlapping? Okay, so if I look at 20 and 25, do those error bars overlap? So I put this little colored here just to be in the same like stretch as this one was. You can kind of see, yes, look, oh, 20 overlaps the error bar. So this data is not statistically significant, so it doesn't show us that it is actually from that temperature, right? But if we look here at 30 degrees, the error bar from 30 degrees does not overlap the error bar for 25. So that's going to show us that 30 degrees is going to be statistically different, right? So this is going to be our 30 degrees where we're going to determine that it is different in terms of the oxygen consumption, okay? So based on the data in table 1, describe the effect of temperature on the rate of the ATP synthesis. So we're looking at this bottom. Oh, I went ahead a little fast. And we're looking at that if we increase temperature, what happens? So an increase in temperature, I go from 12 to 16 to 21. Oh, wow, the number increased. So as the temperature increased, the rate of ATP synthesis also increased. This was a positive relationship where temperature and ATP synthesis are directly correlated, okay? And so a lot of students will say temperature causes an increase in the rate. Well, what about the temperature? So you need to give a little bit more information, okay? So you need to, like say, an increase in temperature with an increase of ATP synthesis. You need to talk about how they're both going to be increasing, okay? So next we have based on the data in table 1. Oh gosh, another math question. Ms. Jones, stop giving me so much math. It's okay, y'all. So calculate the average amount of oxygen consumed in nanomoles for 10 milligrams of mitochondrial protein after 10 minutes at 25 degrees Celsius, okay? So we're looking at oxygen consumption. So we're going to look at this first row right here, okay? And we're looking at 25 degrees. So I'm looking at the 16.5 is what I'm looking at, okay? So the units, nanomole per minute per milligram. Okay, so, oh, I just went ahead and showed you the answer. I'm so sorry. So we have 16 nanomoles per minute per milligram. Okay, then we'll do times our 10 milligrams times the 10 minutes. So I have 1,650 nanomoles. Okay, and so that would be our answer for that one. Okay, let's do another one of those. Can you figure out what kind of graph we're looking at? Here I've got 6 different species. I'm looking at the mitochondria at normal carbon dioxide and the mitochondria at elevated carbon dioxide. What type of graph will we have in 3, 2, 1? Yeah, y'all. It's another bar graph, okay? This has to do with because of the fact that we're looking at categorical data, right? There is 6 different species have no connection to each other. So although I have the numbers going 1 through 6, okay, we're looking at each individual one, right? I'm looking at how is one different in the normal versus the elevated, okay? So we're looking at 1 and 1, okay? So do not try to make this into a another type of bar, like a line graph or something, okay? So this question, actually, they asked them, like, which of them are going to be statistically significant, and the answer is actually all of them. So if you notice that all these error bars do not overlap, and I think that threw a couple of students through a thing. I only pulled the previous FRQ, so no, there's no log, there's no dual Y, and there's no box and whisker. So determine which of them showed difference. Oh, I was ahead of myself, sorry. All species, yay. Okay, so I got another one for you. Which one is this? I've got strains of corn. I've got 6 different strains of corn. I'm looking at the number of double strand breaks, and I'm looking at the number of crossing over. What kind of graph is this going to be? I'm going to give it to you again. 3, 2, 1. It's a scatter plot. I know you told me bar. I know you probably told me bar. So this is going to be a scatter plot. And so you can see that we plotted each of the data points. So I just went and put, okay, well, here is my 19.5. I went above by 0.5 and below by 0.5 to show me my error bar, okay? And then I put each of the different double strand breaks on my X. And it actually, this year, they pre-told them what was on each of the axes, okay? So in this one, like, you kind of could tell what kind of graph it was going to be in terms of that. But they did label each of their data points. There were some students who did it with like symbols. So either one was perfectly fine. So yeah, I know. Sorry, bro. So next one we have, okay, now we're looking at that we have 8 different individuals and we have our blood glucose level. Why is this scatter plot? Because of the fact that I'm comparing 2 different data sets and I've got individualized data, okay? So each of these data points don't really correlate with each other, okay? And so I'm doing a scatter plot to see their overall relationship between the number of breaks and the crossing over that happens between them, okay? So let's go back to this one. So give me your answer in 3, 2, 1. It's a bar graph. I know you were expecting something else, but no, it's another bar graph. So this one, we're going to do each of our different individuals. They don't have any correspondence to each other, okay? But this one brings up our good point, okay? So overlapping is fine. Anyways, so in this one, your teachers may have taught you something called a modified bar graph, okay? And so a modified bar graph is not accepted. So you may be going through some of the old FRQs and you're going to see something called, it'll be like a bar graph or a modified bar graph, okay? So here's the thing. Modified bar graph is not accepted anymore and some students keep freaking out, okay? So I'm taking a moment, we're going to look at what a bar graph looks like, okay? This is a modified bar, okay? It looks like a scatter plot, don't get me wrong, okay? It does look like a scatter plot, but this one's supposed to be a bar graph. And so because of the fact they just put that point and they put lines over it, it looks like that we are like trying to do everything in the middle, okay? And that's what I think happened, is that students were trying to figure things out, okay? And they're just like, I'm going to throw this out, and they're going to accept something, okay? So this is a modified bar graph. This is not acceptable. Do not do this unless it's a scatter plot, okay? If this is a line graph, you will connect the dots, okay? It's very simple, okay? So your bar graph should look like this. Your bar graph should not look like this, okay? So this is not acceptable form. Do not do this, okay? So here, let's do one more. I think this is our last graphing one, and then we're going to get into some content. And oh my God, it's already 46 minutes, okay? So here we're looking at data. We're looking at over some time. I've got some species A and B grown separately and then grown together. What kind of graph am I going to make in 3, 2, 1? Yeah, it's a line graph, y'all. I did not. Hey, you said I was having villains with y'all. No. This is a line graph. I got you one. Don't worry. I went back into 2019 for you just to get you a line graph, okay? So you can kind of see the line graph. They also pre-labeled 2 of them. In terms of your exam, it's just going to be a empty graph because I have to use the same one for everybody. Do not connect the dots on scatterplot, but you will draw a trend line in there, okay? Okay, so now we start the speed review, okay? And when I say speed review, I mean, like, I got to talk faster than this because I got to do 8 units in an hour and 15 minutes because I got somewhere to go, okay? So if at any point you're like, hey, I need some, more information. Like, I can't do Mr. Jones' speed thing. Plus, I'm going very quickly through the information. I'm going to, when I say speed run, I'm going to speed run. I may not say everything on the slide. Like, I am just going. A line graph is going to be linear data. A bar graph is categorical data. So this QR code right here, if you scan it, it takes you to the page that has all of the sessions, including today's session. Those of you like, hey, how can I get to this live stream later? That's how you can get to the live stream later, as well as you've got the PowerPoint for today's thing there too. If you're like, hey, no, I need like a 3-minute or over something. I also have this will link to the page that has where you can get to all the TikToks. You're going to have to go to like, oh, I need unit 2, I need unit 4, but like you can get to them from this page. Okay. So that gives you a second to be able to do your little quick QR code. And I'm going to move on in like 3, 2, yeah, bye. So unit 1, okay, so my goal was to get 3 slides per unit. I don't think that happened in the later units, but I did my best, okay? You can always tell what unit we're in because at the very bottom right here, it'll say what unit we're doing, okay? We're going to go through 4 units. We're going to do some quick questions, and then we're going to do 4 units, and we're going to do quick questions, and I'm hoping to be out of here by 5, okay? Let's do this. We got this. I believe in us, okay? So Water is important. Okay, the things we need to make sure we understand is that water has hydrogen bonds. Hydrogen bond is this little dotted line you see in between there is an attractive bond. Okay, because of the fact that the oxygen is electronegative and the hydrogen is not as electronegative, the oxygen is going to pull more of it in this polar covalent bond that is between them. That causes hydrogen to be partially positive, and this oxygen becomes partially negative. And because of the fact that one's partially positive, one's partially negative, there's an attraction between them. That attraction is the hydrogen bond. That then leads us to many different properties. Okay, one of the properties would be, of course, our cohesion. Cohesion has to do with water molecules attracted to other water molecules. Versus adhesion has to do with water molecules attracted to other polar substances. So it's going to like climb the sides of the capillary tube, if you may, or if you've ever seen a meniscus, it kind of climbs the sides of your like graduated cylinder. Okay, and so we also have evaporative cooling. So if you're hot, right, you're sweating. The reason why you're sweating because of course you're hot, duh. So the water is going to be on your hot skin, right? And so the heat from your skin is going to heat up the water and it's going to cause those hydrogen bonds to break, right? Which then causes your liquid water to become a solid water. A liquid water become a gas water, which then kind of evaporates and it takes that heat away from you, okay? So high specific heat has to do with that it takes a very large amount of energy in order for us to y'all are funny. In order for it to change its temperature, okay, that is allowing good for like our coastal regions. Okay, in our coastal region, we're going to see that it's going to be a lot cooler there because it's absorbing all that heat. Okay, versus when we look at like in the desert area, right? All during the day it's really hot, and at night it's really cold, right? Because the fact that the ground can't really absorb that heat really well and just dissipates and goes out, right? Yes, you can skip an answer if you don't know it. Oh, so in terms of our breaking and forming bonds, hydrolysis is when we're going to add water. Hydro meaning water, lysis means to break. So we're going to add a water molecule. By adding that water molecule, we're going to break a bond. So in this case, we went through hydrolysis, looking at a dipeptide. We broke it into two different amino acids. I know this is an amino acid because of the fact that I have a amino group here and a carboxyl group there, and the bond that was broken was a peptide bond. Okay, and then I can do the reverse of that, looking at dehydration. Dehydration. If you're dehydrated, as you just told me, take water, Miss Jones. Breathe. Right. Dehydration means that you've removed the water. Right. There's no water. Right. And so in a dehydration reaction, we're going to take out the water. So if you see here, we have a hydrogen and an oxygen. We have a hydroxyl group. We have a hydrogen, okay? So OH and H comes out together, okay? And that makes water. So the water comes out. And then what's going to happen is that we're going to, no, these slides are not new. So we're going to have this like emptiness, right? They need a bond, right? So then you're going to attach the bond between the two of them. And now we've formed that new peptide bond, okay? And notice real quick that the peptide bond is between the carboxyl group and the amine group of the next one. We got that N terminus and our C terminus. That's going to be very important as we go into this future stuff, okay? So in terms of monomers and polymers and what kind of bonds are they? So a monosaccharide is the monomer, a polysaccharide is our polymer. The bond that's between them is a covalent bond. It's a glycosidic linkage, but I think all you really need to know about is a covalent bond. In terms of our nucleic acids, we're looking at that we have a nucleotide as the monomer, and the polymer is a polynucleotide or a nucleic acid, so like DNA or RNA, and then the bond between them is a covalent bond, okay? It's between the peptide of one and then the hydroxyl of the next, okay? And so this will give us a phosphodiester linkage. Again, you don't need to know that. You just need to know covalent bond. But the amino acid is going to be the monomer that we have, as we said right here, in our protein, okay? And so the polypeptide would be the polymer or protein, and the bond between them is a peptide bond, okay? I would know peptide bond. Like if I knew one of them, I would know peptide bond. Yes, it is going to be a covalent bond, but it is called a peptide bond, and we definitely need to know that, okay? So I made this cute little chart. I did my best. I had to cut a lot of information to make the chart, but I did my best to make the chart, okay? I don't know why I spent 9 hours building all this, but I did, okay? Anyway, so carbohydrates. We need to know what elements are in it. Carbon, hydrogen, and oxygen. It's about a 1:2 ratio. Okay, as we said before, the monomer is the monosaccharide, the polymer is the polysaccharide. Important information. The cellulose makes up our plant cell walls. Chitin makes up the fungal cell walls, okay? Starch is going to be our plant storage molecule, and it's very linear chains, okay? Versus glycogen is the animal storage, and it's going to be in your liver, and it's branched, highly branched, okay? The lipids, we have carbon, hydrogen, and oxygen. And then in our phospholipids, we do have a phosphate. So I have this and phosphate here, okay? They don't have a, that is definitely in the wrong spot. There's no, actually this is in the right spot. Anyways, okay, we don't have a monomer because there's no repeating structure. Monomers are repeating structures. And so there's no repeating structure here, okay? And so the thing that keeps them all together is the fact that they're all nonpolar and they're all going to be hydrophobic, okay? There are 3 different types that we have. We have our fats, we have our steroids, and we have our phospholipids. These are not polymers. I just didn't really have another spot to put them, so that's where I went. But the fat is going to be 3 fatty acids and a glycerol molecule. A phospholipid is 2 fatty acids, the phosphate group, and then, again, that glycerol molecule, and then the steroid is going to be 4 fused rings, okay? Things to know, saturated is going to have no double bonds, so it tends to be solid at room temperature, versus unsaturated is going to have double bonds, so it usually tends to be kind of an oil at room temperature because of the fact that it has those kinks from that double bond, okay? Phospholipids are going to be making up our plasma membrane, and then steroids are going to be important because they're part of our nonpolar signaling molecules. So they can allow to get through that plasma membrane because the plasma membrane is nonpolar, and it gets right through it, right? Nucleic acids, we have carbon, hydrogen, oxygen. I made these a different color just to kind of symbolize them. There's nitrogen in our nucleic acids, and there's a phosphate. The phosphate's right here because in every one of our nucleotides, you've got a phosphate group, you have a pentose sugar, and a nitrogenous base. The 5' N is going to be wherever your phosphate is, and 3' N is wherever your sugar is. Basically, is the hydroxyl group on that sugar. Okay. The polymer we said is nucleotide, polynucleotide. Nucleic acid would be DNA and RNA. Purines are going to have the double ring, so small name, big structure. Permides are going to have the single ring, so big name, small structure. The base pairing, adenine goes with thymine, adenine goes with uracil, cytosine goes with guanine, and then DNA we're going to see deoxyribose and thymine, as well as RNA we're going to see ribose and uracil. Sometimes they try to throw you off and they'll ask you about like DNA and they'll put an answer that has uracil in it. They'll say, you know, what percent of it is uracil of this DNA molecule? Like 0%? Come on. Give me a hard question. So proteins, that would be carbon, hydrogen, oxygen, nitrogen, and sulfur. Okay. So again, we have our N-terminus side and our C-terminus side. And so the monomer is an amino acid. It's made up of the amine group, the carboxyl group, our R group, that's our variable group, as well as this hydrogen. And so these R groups can make it so that it's either going to be polar or nonpolar. So if that R group is a polar group, it's going to fold outward toward the kind of environment. Okay. If it's nonpolar, it's going to fold toward the inside away from the environment. Okay. We have 4 different levels of folding. So in our primary structure, we're going to have our string of amino acids. Our secondary structures are alpha helix, beta pleated sheet. Our tertiary is where we're going to look at our R groups. And so we're going to have any type of bond that you see with that R group. The beta is like kind of like a wave, and the alpha helix is like a spiral. Okay. And so with our tertiary group, that's looking at the R groups and how those are interacting together. And then with our coordinate structure, we have multiple different polypeptides. So like hemoglobin is made up of 4 different polypeptides. And so those 4 different subunits are interacting together. And that's what makes that coordinate structure. So there's the end of Unit 1. Unit 2, we've got all these different organelles. Okay, so in terms of this one that we see, we've got the smoothie R. The job of the smoothie R is for detoxification of our cells. It helps with our lipid synthesis. Ribosomes, y'all, last year the very first question on the FRQ was, what was the function of ribosome? Do you know how many students sat there staring at it going, I don't know. Okay, y'all, it is going to synthesize proteins according to a messenger RNA or mRNA sequence. Okay, RuffER is going to allow for compartmentalization as well as protein synthesis. There's going to be membrane proteins. There's going to be ribosomes that sit on the membrane of the RuffER, and so it allows you to make membrane proteins as well as protein for secretion. Okay, mitochondria is going to provide different compartments so that we'll be able to go through the process of aerobic respiration within the mitochondria. Keep in mind that that's where we have our Krebs cycle, as well as that's where we have our oxidative phosphorylation. Okay, the Golgi complex. This is going to allow us to correctly fold and modify the different products from our RuffER, and it also packages our different proteins for trafficking. Our lysosome is going to allow for intracellular digestion, so it's going to have hydrolytic enzymes. The hydrolytic enzymes will then break down each of these different molecules and kind of get rid of them, right? Our vacuole, there's 3 different types that we talk about. Our central vacuole, we're going to find in our plant cells. Those are going to help to maintain our turgor pressure. So they'll be full of water and they kind of push up against the cell wall and it pushes back and that kind of gives it that kind of turgid feeling. Our contractile vacuole is going to kind of contract, which then pushes all the excess water out. So there's water rushing into these like, I don't know, plasmodia or something. And so because of the fact that they live in a freshwater environment. And so we need to have the contractile vacuole pumping to then push that water out so that they don't burst, right? So then we have our food vacuole. Food vacuole is going to be made when we go through phagocytosis. Phagocytosis is where we're going through cellular eating. So we go and we engulf something, right? And we bring it in. The thing that is being formed when we bring it in is called a food vacuole. That food vacuole is then going to fold, is going to bind with our lysosome, and the lysosome will then break it down, okay? And then, of course, when we look at our plant cells, we have a chloroplast. That is the location that we're going to see for our photosynthesis. The comments some of y'all have. So membrane transport, okay, they're going to oftentimes ask us about how things are getting across our membranes, right? And students sometimes screw this up, okay? And it happens, we're good, no worries, okay? This is our membrane. As you see, our membrane is made up of phospholipids. Sometimes there are going to be channels in it that can help the things move across. So this is the channel protein, this is the carrier protein, okay? So the thing is that the outside, this little head right here, this is going to be polar. It's hydrophilic, okay? While the inside part right here, the interior of the membrane, okay, this is going to be nonpolar, hydrophobic, because this is made up of these fatty acid chains, right? Okay, and so because of this, we find that overall the membrane is nonpolar. okay? And so if we have something that is nonpolar, carbon dioxide, oxygen, nitrogen, steroids, these are nonpolar. They're small, nonpolar. They can get straight through it, okay? They can go through something called simple diffusion. They don't need transport protein. They don't need anything but a gradient, okay? So they're going to go down their concentration gradient. They're going to go from a high concentration to a low concentration. As you see, there's a high amount of these little blue dots, and there's a low amount of blue dots on this side, and so it's going to move across by simple diffusion, okay? Sometimes we have some of our small molecules that are like water or ammonia that can leak through the membrane. They're going to go through very, very slowly because they're polar and they're not going to be able to get through as easily, but they can leak, okay? Now, in terms of facilitated diffusion means that we're going to have some help, right? A facilitator helps you in a discussion, right? So facilitated diffusion means that there's some type of assistance that's going to facilitate it to move it across the membrane, right? So this right here is a channel protein. A channel protein is a transport protein that's going to allow the movement from one side to the other, okay? So let's say this polar substance, I don't know, sodium ions, okay? So this sodium ion is going to bind into this channel protein that's going to be specific to it, right? This is going to have the right size, shape, charge, all the good stuff to allow this material to pass straight through it, okay? And so it's going to go from high concentration to low concentration, high to low, as we saw before. So it doesn't require any type of energy. But it does need a transfer protein to get from one to the other side. So that's what we call it facilitated. So like water, which you can use aquaporin. We have like sodium and potassium and calcium. These are all charged particles. And so because they're charged, they're not going to easily get across that membrane, right? And as I already said, so we can either have a channel protein or a carrier protein to do that, okay? Now, active transport, okay? So both simple and facilitated were both passive transport. They did not require any type of energy, right? But active transport does require energy. We need to have some type of ATP, as you see right here. We're going to hydrolyze the ATP, and by hydrolyzing the ATP, we're going to phosphorylate that protein. And by phosphorylating protein, you charge it to do work, right? And so by allowing it to do work, by giving it that energy, okay, it's going to allow to pump material against the concentration gradient, right? We're going to take something from this low side and we're going to pump it across to where there's a high amount, okay? So when I say high and low, I'm talking about the concentration. This is a high concentration. This is a low concentration. So in simple diffusion, we're going from high concentration to low concentration. We go down the concentration gradient. But here I have a high concentration. Here I have a low concentration. And in active transport, I'm going from low to high, and that's going to go against the gradient, and so that's going to be requiring some type of ATP, some type of energy, okay? As I said, it's going against gradient. It requires some transfer protein, so usually it's going to be a carrier protein. A channel is just literally just a passageway, so that's not going to work, okay? And then we also have bulk transport, so exocytosis is going to be like when something's leaving the cell, versus with endocytosis, something coming into the cell. So here you're seeing different forms of endocytosis. This is phagocytosis, so cellular eating, as we said before. We're going to form our food vacuole. Y'all, we're going to form a food vacuole. The food vacuole will then fuse with our lysosome, okay? Penocytosis, this is going to be cellular drinking. So when you drink a lot, it makes you pee, so it's going to be cellular drinking. We're just going to try to get some extracellular fluid in. And then with receptor-mediated endocytosis, this is going to involve where we're going to have there's some type of ligand that binds that then causes it to bring in. Bulk transport is necessary. I don't know what blood cell is going to engulf a bacteria to help fight an infection. That's bulk. It also brings in like some of our like amoebas and whatnot need to eat that way. Okay. So with osmosis, okay, then we have different types of solutions. We have hypotonic and hypertonic, okay? So think about a friend of yours, right? Are you taller than your friend or are you shorter than your friend, right? And then think about someone else that's on the other side, right? So if you were, let's say, for example, I am taller than my children and my husband is taller than me, okay? So if I'm in comparison to my kids, I'm tall. But if in comparison to my husband, I'm short, okay? So that's what we're going to see here with these solutions, okay? Is that we're going to have two different solutions on the side of a membrane, okay? On one side of the membrane, there's going to be a high concentration of solutes, which means there's a low amount of free water. And then the other side, we're going to have a high amount of free water and a low amount of solute, okay? And so the solution that has a low amount of solute is going to be called hypotonic, and the solution that has a high amount of solute is going to be hypertonic, okay? So we're going to go from a, where there's a high amount of free water to where there's a low amount of free water. Give me 1 second. Boys! Shh! So what we're going to see is that we're going to go from a kind of where there is a low amount of, where there's a high amount of free water to where there's a low amount of free water, okay? And the water is going to move from the hypotonic to the hypertonic, okay? Isotonic means that it's going to be equal, okay? We have equal amounts on both sides of the membrane, okay? And so Due to this, we're going to see that it's going to go equally to both sides, okay? And so if we were kind of to look at it in like a visual format here, this would be a hypotonic solution. There's not any solutes in it, so we're not really surrounding any of those solutes. Whereas over here, it's going to be a hypertonic solution, so the water's going to move from the hypotonic to the hypertonic. If we were to take an animal cell and put it into a hypertonic solution, it's going to lose all of its water, so it causes it to crenate or shrivel. If you put a plant cell in a hypertonic solution, that means that it's going to go through something called plasmolysis, okay? I didn't clock them. Isotonic has to do with that they're going to be kind of equal on both sides, we said. And so we're going to see that this is what it is normal for an animal cell. It's going to equally go back and forth. Versus in a plant cell, it's going to be flaccid, so it kind of is limp. Versus in a hypotonic solution, if all the water is rushing into my animal cell, there's no cell wall and it's going to burst, okay? And so that's going to be a problem, okay? Versus in our plant cell, we want that water rushing in because that's going to cause the turgor pressure, which kind of makes it turgid and kind of makes it kind of have that pressure to it, which is best for it. Do you need to know the different water potential stuff? Yeah. I cut it in terms of time because, well, we only have so much time, and I right now have an hour to get through, 6 more units plus do all the practice questions. Okay. So let's get into unit 3. So enzymes are a biological catalyst. They're going to speed up chemical reaction. The reason why they speed up chemical reaction is because of the fact they're going to decrease our activation energy. So this right here is our activation energy. The purple loop is or the hump is what we really kind of need in order for the reaction to take place. But the green is kind of showing us with our enzyme, it decreases the amount of energy that's needed. And so because of the fact that we've decreased the amount of energy, it's going to go a lot more frequently. Okay, it's going to go faster. Okay, and then so as you see, we got this delta G. That's our change in free energy. So in this reaction, we're looking at an exergonic reaction because it's releasing the energy. Okay, so we go from a high amount of free energy to a low amount of free energy. Okay, because of the fact that it's releasing that out into the environment. So there's that. Okay, enzymes are proteins. So everything you know about enzymes, okay, applies to, everything you know about proteins applies to the enzymes, okay? So they're not consumed by the reaction. They will work forever and ever 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 and ever. Never. They will work forever, okay? And they have no effect on the Gibbs free energy. They're not going to make a reaction that wasn't going to take place, okay? We're still going to start with the same amount of energy and end with the same amount of energy. We're still losing the same amount there. Like, that's not changing. The only thing that changes right here is this activation energy, okay? Now, there's a lot of different environmental factors, okay? You do not need to know delta G, but you need to understand what delta G is, okay? You need to understand that it's free energy. You need to understand that you need that free energy for work, right? You need an exergonic reaction to fuel an endergonic reaction. Endosymbiotic has to do with the theory that a mitochondrial chloroplast was engulfed. So concentration of our substrate in our enzyme, okay? So if we have more of our substrate, then we're going to reach this point where our enzyme is going to get super saturated and it's going to work as fast as it can, but it's going to kind of level off in terms of its rate, okay? In terms of inhibitors, we'll talk about those in a second. Temperature, if we have an extremely high temperature, it's going to cause your enzyme to denature, right? It causes the protein to unravel. If you have a low temperature, your reaction rate is going to slow down only because of the fact that the kinetic energy slows down. If we have a little bit of an increase in temperature, not super amount, but a little bit of energy, okay, a little bit of increase in temperature, it's going to speed up our kinetic energy. Things just move a little bit faster. And because things move faster, we're going to see that these bonds are going to break, right? And so in terms of pH, if we go one outside of whatever the optimal is, it's going to denature either way, okay? So denaturation just means that it unravels, it goes back to that primary structure, it breaks those hydrogen bonds that are in the secondary structure, it's breaking all the bonds within those R groups, okay? And so it's just going to unravel back to that primary structure, okay? Gibbs free energy is just the energy that's available to do work, okay? So as we said, if you get outside that temperature, that pH range, it's going to disrupt the hydrogen bonds. Inhibitors, there's two different types. We have competitive inhibitors and we have non-competitive inhibitors. Competitive inhibitor is going to bind to the active site. So the active site is where we're going to see that this material is going to bind. Like the substrate binds to the enzyme at the active site, okay? Now, if the substrate is binding here and the competitive inhibitor is binding to there, that competitive inhibitor is going to be like fighting for that position, and it's going to slow down the reaction rate because of the fact that this competitive inhibitor is binding to it instead of the substrate, okay? A non-competitive inhibitor, it binds to an allosteric site. It binds to a site somewhere else on that enzyme, and because it binds to somewhere else on the enzyme, it's going to change the active site. I tell my students, anytime you bind something to a protein, it always changes shape, like always changes shape, okay? And so that's what's happening is we bound something to the protein changed shape, and because that shape changed, now my substrate can't bind to the active site, okay? So photosynthesis, y'all, You freak out every year, okay? You need to have big picture. What goes in, what comes out, why is it important? Like, where does it take place and why is it important? Okay. So, in terms of our photosynthesis, there's two different steps. We have our light reaction and our Calvin cycle. The light reactions are going to take place in thylakoid membrane or the grana. Okay. So if you think about the chloroplast has those little green like sacs inside of them. Those are your thylakoid sacs. Okay. And the stack of them together makes the grana. Okay. So in the membrane of that is where we're going to see the light reactions. We're going to see that in photosystem one there is chlorophyll A. Okay. We're going to start out with electrons. Electrons are going to come from water. So right here you see we have this water molecule. It's going to go through something called photolysis, where we're going to break that water. And by breaking that water, we can release oxygen out, and then we get the electrons. Electrons are going to go here to P680, or so they're going to go here to that chlorophyll molecule, okay, to regenerate the electrons once it gets lost, okay. And we also have photons in terms of light energy. So the light's going to shine down on the photosystem. It causes those electrons to get excited, and then we're going to have the energy come down to this reaction center site right here, okay. And then because of the fact that that energy goes up, right, boom, it comes up to a higher energy state, it's going to get stolen by a primary electron acceptor. And so what happens is it gets stolen, and then it goes down this electron transport chain. So as it goes down the electron transport chain, it has a whole bunch of energy. So as it goes down that chain, it releases a little bit of that energy, and as it releases energy, it pumps a proton across the membrane. So we're going to see that these protons get pumped across the membrane, so they're going to go from a low concentration to a high concentration. We're pumping them, okay? We're doing some form of active transport in a way, right? And the energy is coming from the electron releasing that energy, okay? And so what ends up happening is at the end of this, that electron ends up in our P700, our PS1, our photosystem one, to regenerate the electron that one loses. And then we're going to see is that it will go over here and get taken by NADPH, which I made it so that this one pops in. So ignore this arrow for a second. So this is where NADPH is, and we're going to carry the electron and take it over to the Calvin cycle, okay? So in linear electron flow, we go through photosystem 2, we go through the electron transfer chain, we go to photosystem 1, and then we go to NADPH, okay? The P stands for photosynthesis, and the H stands for holding. That's a lie, but you can deal with it. It's okay. And then cyclic electron flow just means that we're going to cycle back from here, okay? So we're going to start with photosystem 2, and I'm sorry, photosystem 1, and we're going to cycle back over here to the electron transport chain so we can generate that proton gradient. That proton gradient is used to make ATP. Okay, so it's not actually this electron transport chain itself that makes the ATP. It's that forms that gradient. That gradient then goes down its like gradient through ATP synthase to then be able to synthesize that ATP molecule. Okay, so the ATP and the NADPH are then going to go over to the stroma. The stroma is going to be the jelly substance that's in our chlorophyll. I'm sorry, chloroplast. And so it's like the cytosol of the chloroplast. Okay, and so we're going to start with 3 molecules of carbon dioxide, 9 molecules of ATP, and 6 molecules of NADPH, and we're going to make something called G3P. 2G3Ps come together. The product of water is somewhere else. I can't remember right now. Not important. And so what's going to end up happening is that we're going to make G3P. The 2G3Ps come together, which is then going to make the glucose. Okay. So they basically will then send back the NADP plus, and they'll send back the ADP for it to then get another thing, okay? So in terms of cell respiration, we're going to have 3 steps: glycolysis, Krebs cycle, and oxidative phosphorylation. Glycolysis takes place in cytosol, so all organisms can go through glycolysis. It starts out with glucose, and it ends up with 2 pyruvates, 2 NADHs, and 2 ATPs. Now notice this is not NADPH, this is just NADH. The H still stands for holding, meaning that we're going to hold the electron and take it over somewhere else, okay? And so in glycolysis, glyco meaning sugar, lysis meaning to break. So in the sugar splitting step, we're going to break up this glucose molecule into pyruvate, and then we're going to release the electrons in NADH, and then also we make 2 molecules of ATP. We technically make 4, but we use 2 of them, so whatever. Anyway, so Krebs cycle. Krebs cycle doesn't take place in the matrix. So if you think about like the outside of our mitochondria, there's this inner membrane called the cristae. On the inside of that is where we're going to see the Krebs cycle. That's the matrix. Okay. So the Krebs cycle, we're going to finish the breakdown of our glucose molecule, and so we're going to start with acetyl-CoA. We make 2 molecules of carbon dioxide, 3 NADHs, 1 FADH2, and then 1 ATP. Okay. And so we are completely done with glucose at this point. All we have is the electrons from it. Those electrons are in the form of NADH and FADH2 to take those over to our oxidative phosphorylation. Okay, and then in oxidative phosphorylation, there are 2 steps to this. We have electron transport chain, and then we also have chemiosmosis. Okay, so in oxidative phosphorylation, in electron transport chain, we're dropping off the electrons here, and then they're going to go down the chain. And as they go down the chain, again, they're dropping in energy, and they're going to pump that proton across that membrane. As they pump the proton across the membrane, that gradient is then going to be used by ATP synthase. Okay, so this right here, this protein, it tells you what it does. ATP synthase is going to synthesize ATP. Okay, so it's then going to go down this concentration gradient, turning that turbine, so that we can then make that ATP. Okay, and so we start with NADH and FADH2 with our electrons. We make the ATP, and we have electron transport chain and the chemiosmosis. Okay, notice at the end electron transport chain where we have oxygen. That tends to be a question that comes up a lot about like metabolism and like the oxygen. And so there's going to be an increase in the oxygen usage whenever there's an increase in metabolism because of the fact that we're going to use more oxygen because it's the final electron acceptor. Now, if we happen to not have a mitochondria, or if we are backed up, there's some process that's not, we don't have enough NADHs or NAD pluses, okay, then we're going to go through something called fermentation. Fermentation is going to go through glycolysis, and then we need to regenerate the NAD plus, okay, and so it's either going to be alcohol fermentation or lactic acid fermentation, and it's just going to regenerate, okay. They are probably not going to ask you about C4 plants because those are not in the curriculum, but they can ask you about C3 plants, which is normal plants, and cyclic AMP is a secondary messenger, okay. So in terms of unit 4, this one has to do with our signal transduction. Okay, so we have 3 steps. You have the reception, transduction, and the response. Reception is where we're going to receive that signal. So as soon as the ligand, our signaling molecule, binds to the receptor, it then causes it to change shape. And so that is the first step of transduction. So in terms of our ligands, they can either be steroid or they could be hormone, protein hormones. A steroid is going to go straight across the membrane, which means the receptor is going to be on the inside of the cell, versus a protein hormone, its receptor is going to be on the membrane. So if it's a polar ligand, it's going to be on the membrane. If it's a nonpolar ligand, it's going to be on the inside in the cytosol. Okay. So next up we have is transduction. Transduction has to be how we relay. How do we get it from reception to the response? Okay. So the transduction is going to be where we're going to go through a phosphorylation cascade. So we have these protein kinase molecules. Protein kinase's job is going to be to phosphorylate. Okay, so it's going to add a phosphate to a protein. Okay, and so we go through this relay of phosphorylating all these relay molecules. Okay, we also can have secondary materials. So like calcium can be released, and as calcium is released, it then goes and binds to then cause something. This is what happens in our muscle contraction. And then cyclic AMP, as we saw with cholera, is going to be made by the adenylyl cyclase and then causes the activation of protein kinase. But you did see that diagram now that I'm thinking about it. I didn't show it to you. Sorry. And then the response, these are the things that are in the CED. We need to know that it can cause the cell to grow, cause secretion of material. It can change gene expressions or turning on or turn off the gene, so it acts as a transcription factor, or it can act, alter the function of it, or it can go through apoptosis. Okay. So in terms of feedback, we have positive feedback and negative feedback. Negative feedback is going to bring us back to homeostasis, okay? The product is going to come back and it's going to inhibit the process. It can either inhibit the process by turning off the enzyme, so it can inhibit an enzyme. And if you inhibit the enzyme, then the whole pathway slows down because we're not having the thing that's supposed to speed it up. I think that there's still a typo. It's a typo. This is messenger. Sorry, that's a typo. This should say secondary messengers, not secondary hormones. Sorry, y'all. So with our negative feedback, it also can come back and it can turn off a gene. And so if I don't make the enzyme, then of course I'm definitely going to slow down the decreasing, the making of that certain pathway, right? Positive feedback is going to be the product going to come back and it's going to amplify, okay? It's going to make it so it keeps building up, okay? And so whatever the product is comes back and it keeps the process continuing to go, okay? So for some reason, they switch out between mitosis in one unit and meiosis in the other. So we're going to go through the cell cycle and mitosis, and then we're going to take our break, and then we're going to go through meiosis, okay? And so for the cell cycle, we've got interphase, which is G1, S, and G2. This is kind of the in-between the cell division process. This is where it's kind of trying to be a normal cell. And so G1 is where it's metabolically active. It's going to duplicate its organelles and the cytosolic components, okay? There is a checkpoint in the G1. The G1 checkpoint is going to make sure that should I be dividing? Should the cell go through division? And so there's going to be, do you have a growth factor? Does a growth factor bind to then cause you to go through this process, okay? Are there enough resources? Is there enough resources for my cell to be dividing? I don't know about that, okay? And then it's going to check for DNA damage. If there's any damage in the DNA, we do not want to replicate our cells. So it's going to check for all that. If it doesn't pass this, it goes out into G0. G0 or G0 is going to be where it's no longer dividing. The cell is just being a cell. It's just chilling. You do not need to know whatever enzyme you just mentioned. S is going to be our synthesis, okay? This is what we're going to have where we're going to replicate the DNA. We go through DNA replication. We synthesize a copy of the DNA, okay? And so it forms those sister chromatids, okay? We have no additional genetic information at this point, okay? We just made a copy of the genetic information we already have, okay? And then we go into G2. G2 is where we're going to synthesize our proteins. We make a large amount of ATP, and then our centrosomes are going to replicate in preparation for division, okay? This is going to have a checkpoint. The G2 checkpoint is going to make sure that did all the DNA replicate, and then is it damaged? Is there any issues with that DNA? Okay, and so if it detects a problem, that's where it's going to halt it. It probably gets the P53 to kind of come in and hold the process. We have like the cyclin dependent kinase and the kinase molecules building up in this place. The cyclin is a type of cyclin dependent kinase. Oh, sorry, cyclin is what's going to bind to cyclin dependent kinase. Anyways, and so we're going to see the build of the cyclin. If it gets to a certain level, it then goes through that process. And that will get us into the M phase. M phase is going to be our nuclear division. So this is all interphase right here. So we went through replication before we went through mitosis. And then we're actually going to go through something called like the cytoplasmic division. This cytokinesis will take place after mitosis, okay? And so we have 4 different steps. We've got prophase. Prophase is when the sister chromosomes are going to condense. The mitotic spindle forms. The centrosomes are going to move to the opposite poles. We're kind of preparing to divide. So like prophase. Then we go into metaphase. Metaphase is when the sister chromosomes are going to line up on the metaphase plate or in the middle of the cell, or as the thing said, the equator of the cell. And they are attached by these different spindles. So we've got these microtubules attached to each side that have this little tug of war, and that's what gets them to be in the middle. Okay. Then we go into anaphase. Okay. But before we can get into anaphase, we have to go through another checkpoint. The M checkpoint is going to make sure, did all of the sister chromosomes, are they all attached to a kinetical microtubule? Are they attached? Because if they're not attached, it's going to lead to something called non-disjunction, and we don't want that. We want to make sure that every cell has equal amounts of chromosomes, equal amounts of genetic information. Okay, so we go through the M checkpoint. If everything is attached, then we go into anaphase. Anaphase is when we're going to see the sister chromosomes are going to move apart from each other and then move to the opposite poles. So the paired sister chromosomes are going to separate, pulling to opposite poles. And then telophase is when we're going to make our 2 new nuclei, okay? And that's when the mitotic spindle is going to break down, and we're then going to form our nuclear envelope. Now, at this point, we just have 2 nuclei. Then we go through cytokinesis. Yeah, I had to give you another jump scare. So cytokinesis is when we're going to split the cytoplasm. So we either going to have this cell plate forming in our plant cells to separate them, or we're going to see the cleavage furrow is going to squeeze and pull them apart into opposite ones, okay? So this is the question that's straight out of the CED. So I went through all of them. Based on time, I'm really worried about our time here. So I'm going to, again, speed run them. So they examined that there's a folded structure of a protein that's suspended in water. There's these amino acids with nonpolar R groups that are buried in the middle of the protein, whereas the amino acids with the polar R groups are on the surface. So they're saying that the polar on the outside, the nonpolar on the inside. Why would this be? Okay, so we have to read through the answer choice and figure out which of these is logical, right? Which of these is a logical reason why we're going to find the nonpolar on the inside and the polar on the outside, right? So polar R groups on the surface can form ionic bonds. Nope, they're not going to form ionic bonds because of the fact that we're looking at polar, which means that we're looking at like hydrogen bonding, right? So polar are too bulky to fit in the middle. They're just as bulky as the other ones. There's a reason for that to be the reason. Nonpolar R groups cannot form hydrogen bonds. Well, if it's nonpolar, then it's not going to have the partial positive and partial negative. So you're right. Hydrogen bonds cannot be formed with the nonpolar R group. And so they then get pushed in the middle of the protein because of the fact that they don't have that ability to make those hydrogen bonds. And then last answer choice, nonpolar R groups from different parts of the protein form covalent bonds with each other to maintain the protein structure. Negative. So protein, a student used a microscope to observe a slide of red onion cells that are suspended in a sodium chloride solution. They then added 15% sodium chloride solution to the slide and observed the changes that occurred. The observations are represented in the figure, okay? So here we can see that they had 1% and it's like full of something. But then when I put in the NaCl, it looks like it's been like scrunched up. So which of the following most explains the changes in the cell? Well, if I'm increasing the concentration outside the cell, that makes me think that there must have been a hypertonic environment outside, so the water is rushing out of the cell, and that's what's causing my cell to kind of go and squivel up, right? Which of the following explains the changes? The degradation of DNA in the nuclei of the cells. I do not see that happening. Lysis of chloroplast. Again, why is the chloroplast breaking up? The movement of water from the central vacuoles of the cell into the solution. Yep, so water is moving out of the cell, and because the water is moving out of the cell, that's causing it to swamp up, right? And the movement of sodium chloride from the solution in the cytoplasm of the cell. If something was moving into the cell, we wouldn't see the thing swamping up, right? I don't know. I'm hoping it's over soon. So the enzyme trypsin aids in protein digestion in the small intestines. The relative activity of trypsin at the different pH levels is shown in the figure, which following best explains the activity levels of the trypsin shown in the figure. So here I can see the activity of it is a low activity at a 5, 6 is starting to go up. 7's good. 8 is where it's the highest it's possibly at. And then it goes back down again. So that kind of makes me think that like this 8 range must be its optimal range, and so there must be something happening. And we know that if a protein ends up being outside of its optimal range, it's going to denature, right? And so let's see if any of these talk about denaturation. The small intestine releases inhibitor molecules that blocks the activity of trypsin unless it's at optimal pH. What? I don't know anything about the small intestines. Anyways, the number of effective collisions between trypsin and a substrate increase at higher pH levels. What does that have to do with anything? Anyways, pH values increase, the substrate concentration decreases, leading to an eventual decline in the rate of the catalyzed reaction. So because I have more pH, there's less substrate? That doesn't make sense. At extremely low pH values, the trypsin is denatured. Hey, it says denatured. I got that. Denatured and cannot function efficiently. Okay. So as I said before, sometimes y'all can answer these questions without looking at the answer choices. Okay. So that's kind of what I'm trying to model for you. Okay, is how you would go through these different processes. Okay. So it estimated that the oxygen production first evolved in photosynthetic prokaryotes approximately 2.7 billion years ago. The first photosynthetic prokaryote was presumed to be similar to today's cyanobacteria. We find best supports the claim that photosynthetic prokaryotes are responsible for the oxygen in Earth's atmosphere. Okay, so this wants us to figure out, well, why is the oxygen, right? Why is the photosynthesis how we got the oxygen? Okay, so the light reaction to photosynthesis split carbon dioxide and carbon dioxide and what? No, it doesn't. Carbon dioxide is used in the Calvin cycle, and I'm going to make something with that. So that A is wrong. Let's go to B. The light reaction to photosynthesis split water. Hey, that's right. Introduce hydrogen ions and oxygen. Wait. Okay, I like that one. Let's keep going. The Calvin cycle splits glucose. No, it makes glucose. The Calvin cycle splits water. No, it doesn't. That's light reaction. Well, the only one that was logical to me was B. So, hey, there we go. We were right. We're so smart. I'm so proud of you all. You were just rocking this whole thing. Okay, so on to this next one. So insulin is a protein hormone that's secreted in response to elevated blood glucose levels. When the insulin binds to the receptor in the liver cell, the activated receptors stimulate the phosphorylation cascade that causes the translocation of the glucose transport to the plasma membrane. Oh, what? That makes no sense. Anyways, based on the information provided, which is following best explains the role of insulin in this liver cell transduction pathway. I think I need to read again. I'm confused. Insulin is a protein hormone that is secreted in response to elevated blood glucose levels. Okay, so it's a hormone. So a hormone is a ligand. It's a signaling molecule that goes around the cell. It's like, not around the cell, but like around the body. Okay, cool. And then it binds to a receptor. So there must be a receptor somewhere that it binds to. And then there's a transduction process where I'm going to make a phosphorylation cascade. And then my response is going to be translocation. Okay, so what is the role of insulin? A, acts as a ligand. Hey, I said that. B, acts as a receptor. Can't be a receptor. It talks about it binding to a receptor. That's not possible. It acts as a secondary messenger. That's where this phosphorylation cascade is talking about. And it acts as a protein kinase. Again, protein kinases are going to have our phosphorylation cascade. Okay. So again, we already knew the answer, but sometimes, you got to think through it, right? Cool. So then here we go. I had to make this one a little funky because I couldn't really figure out a best way to put this. Platelets are fragments of larger cells and normally circulate in the blood without adhering to blood vessels. When the wall of the blood vessel is damaged, collagen fibers in the wall are exposed to the interior of the blood vessel. The exposed fibers and chemicals released from the endothelial cells line the blood vessels, attract platelets, which start to form a plug and release other cells as seen in the figure. I thought this animal systems aren't in the curriculum. My teacher told me animal systems were not in the curriculum. Y'all, animal systems not in the curriculum. But this is biology. And what they're going to do is they're going to give us questions that have applicational biology. So you don't actually have to know anything about platelets. You don't actually have to know anything about how the plug is formed, okay? You're going to use the information and you're going to apply that information, okay? So which of all best explains the feedback mechanism, okay? So feedback, we have negative feedback, we have positive feedback. Negative feedback had to do with that the product was going to come back and it's going to like regulate and decrease and bring us back to homeostasis, versus positive feedback was going to amplify some process, okay? So it says that we're looking at that the platelets are fragments of larger cells. When it's damaged, they're going to be exposed to the interior that then releases different chemicals that line it to attract the platelets that then form a plug. And so the platelets are then going to cause even more to be released, so they cause other chemicals to come to the area, okay? So this is an example of positive feedback because a few platelets that initially bind attract more platelets, okay? That sounds like positive. The I have positive feedback because the product, which would be the platelet, causes more platelets to come to the area. And as I have more platelets, that then causes even more platelets to come to the area. I like that one. That sounds good. Okay. Which I have positive feedback again because it results from the interaction among collagen, endothelial cells, and platelets. No, I don't see a buildup happening, so it doesn't make me think positive. This is an example of negative feedback because there's a large clump of platelets can block the blood vessel. I agree it's blocking it, but I mean, like, is that really looking at negative feedback that we're going to try to like? No, that's not seeing what happened. This is an example of negative feedback because an accumulation of our platelets returns to open blood vessels. Now, okay, so A is of course our answer, and you all are rocking this with the A all the way down. We do have some other people saying C and D. That's okay. Whew, we're back to content review, and I have 30 minutes to get through the rest. This is not going to go well. Okay, so meiosis. Meiosis is just like mitosis, but now we're going to go through it 2 times, right? So we replicate 1 time, and then we're going to divide 2 times. The reason why we're going to divide 2 times is because we need to make it so that we have half as much of the genetic information because of the fact that we're trying to make a gamete. Okay, and so in prophase 1, we're going to see our homologous chromosomes. Homologous chromosomes would be having our sister chromosome from our maternal and our sister chromosome from our paternal. That makes our homologous pair or homologous chromosomes. Okay, so when we work through meiosis 1, we're looking at those pairs. Okay, and so with prophase 1, homologous chromosomes are going to pair up and they're going to condense. Our synapsis occurs, and that's when we're going to see crossing over taking place. Okay. So these non-sister chromatids are going to overlap and they're going to exchange genetic information, okay? And so if we were to say, for example, that we've got my paternal and my maternal chromosome, right? And they cross over, what happens is that they're going to exchange genetic information. And so you can make a recombinant chromosome. The chromosome is going to have partial of, the genes from the maternal as well as the genes from the maternal and paternal, okay? And so you can actually get a recombinant chromosome while you have 2 that are still going to be the parental that did not have crossing over taking place, okay? In metaphase, this is going to deal with our homologous chromosomes. They're going to be on the metaphase plate, okay? Similar to how we saw it in mitosis, but now we're just looking at homologous chromosomes, okay? In anaphase, homologous chromosomes are going to separate to go into opposite sides, right? They're going to be pulled apart, okay? And then telophase is going to be where we're going to form our 2 new nuclei, right? And now this is going to already be haploid, okay? I separated maternal was one way, paternal went the other, okay? So this cell over here is only going to have 1 set of each of the chromosomes. There is a duplicate, don't get me wrong. I do have 2 of them all, but they're identical-ish to each other. And so we're going to call them haploid at this point. Then we go through meiosis is the exact same as mitosis because we're going to see that in prophase, we're going to prepare to divide because of the fact that prophase 2, they're now the sister chromatids are going to condense, all that good stuff. Then we move into metaphase, sister chromatids line up on the metaphase plate. In a phase, they're going to move apart into opposite poles. And then telophase, now we have our technically 4 new nuclei, okay? And so this is going to form our 4 cells, okay? So in terms of genetics, say they're going to probably have some type of genetics problem for you, okay? And so they may talk about monohypercross. Monohypercross means it's heterozygous for 1 trait, okay? So we have issues with the math, okay? So if we're doing a monohybrid cross, okay, we would add them together because we've got 1 that's homozygous dominant, and then I've got 2 that are heterozygous, so 3/4 of them would be yellow in this case, and then 1/4 would be green. Now, if I wanted to see yellow and round, then I would do the exact same thing where I see that 3/4 of them are yellow and 3/4 of them are round. So 3/4 times 3/4 gives me 9/16. Then a dihybrid cross, they're heterozygous for 2 traits instead of 1 trait. Okay. And so you can do that math for all of them. And so I personally think that it's better for you to do multiple Punnett squares that are like this, and then you multiply the probabilities in order to get the final answer. Okay. There's a couple different important terms. So like genotype is talking about the actual genes. Okay. If it says it's homozygous, that means that it's same thing. So you have 2 dominant alleles or 2 recessive alleles. Okay. Heterozygous is 1 dominant and 1 recessive. And then the phenotype is the physical characteristic that you see. Okay. Now they may talk about test cross. The test cross means that you're crossing it with something that is going to be homozygous recessive. And so that you can be able to figure out what is the unknown. Okay. Sometimes what they're going to do is they'll have you do a heterozygous with a test cross. And so you'll be able to then look at whether it's independent assortment or not. Okay. Whether those things lined up appropriately. Did I talk about independent assortment? I'm sorry. I think I forgot to mention it. In metaphase 1, we have independent assortment where we can see that those homologous chromosomes can independently align on the metaphase plate. I think I forgot to mention that. I'm so sorry, y'all. Okay, so there's different types of dominance. You can have complete dominance. Complete dominance means that we're going to have one is going to be completely dominant over the other. And so usually what's going to happen is that recessive, it makes a non-functional protein or doesn't make enough of the protein. And so it's going to be able to be masked by that dominant trait or that dominant enzyme, dominant allele, okay? Codominance, you're going to see both of them at the same time. So people that have AB blood, we're seeing codominance. They're seeing the A, glycolipid, glycoprotein, whatever it is, and then also the B at the exact same time on their cell membranes, okay? Incomplete dominance has to do with a blending, okay? So they're making, I don't know, we have red pigment made and we have white pigment made. And so if you have both the red and the white, then you're going to end up being pink. And so it ends up being some type of blended version between the dominant and the recessive phenotypes, okay? In terms of this, because they're both incomplete dominant, okay? So you're probably going to have a pedigree. Pedigrees always show up on the exam. So we're probably going to have some type of pedigree. Okay. And so in this case, we know this was autosomal recessive because of the fact that if we look here, we see these two individuals that they were not affected. And then here they are affected. The reason I know this is autosomal is because this male has to have the trait if it's sex linked. So if it's sex linked, he cannot hide it because he only has one X chromosome. And so there's no way that you would have a X linked recessive in a female because of the fact that he can't pass it on to the offspring, okay, because he can't hide it. So this one is autosomal recessive, okay. X-linked recessive has to do with that it's going to be on the sex chromosome, okay. So oftentimes you'll see an affected mother and then an affected son, okay. The mother can pass it on to her girls, but unless the father also has it, you're not going to see it affected. So that's why you see over here, you've got an affected daughter because the mother and the father were both affected by it, okay. And then we have maternal inheritance. This gets y'all sometimes because they're talking about like mitochondrial inheritance or chloroplast inheritance, okay. This is going to be found in the mother, okay. So the ova is what oftentimes the word that's used is the ovule or the egg. And so in this case, you'll have an affected mother and all of her offspring. So here, the boy, the girl, the boy, the girl, and the boy, they're all affected. But if you look here, this father does not pass on that trait. This father does not pass on the trait, but the mother passes on to all of her offspring, okay. So autosomal was just meaning that it was on one of the autosomes, one of the 22 other chromosomes that you have inside of your cells. Sex linked was going to be on one of the sex chromosomes, so the X chromosome or the Y chromosome. If it was on the X chromosome, it's X linked. If it was on the Y chromosome, it's Y chromosome, Y linked. The chat went silent because we lost 100 people. Maternal inheritance. This has to do with that the allele is located, as I said, on the mitochondrial DNA. Okay, and then linked genes means that it's going to be on the exact same chromosome, okay? If they're close together, they're going to be inherited together, and we call those linked. If they're far apart, due to the crossing over, they may get separated from each other, okay? And so I did want to pull up this pedigree question and just talk through a little bit of the pedigree with you. I know it's a little bit extra, but I do want to make sure we go through this. And so they gave us in the 2021 example, they gave this pedigree. They said that they concluded that it came in this person right here, this 2-2. So what they'll do is they'll name them by like the generation. So generation 1, 2, 3, and then the number of the organism. So this is 2-2, meaning this is the second organism right here. And it says some type of mitochondrial inheritance, right? I don't know why we lost 100 people. So we lost, this is on the mitochondrial DNA, right? And so here we're looking at this person, and we can kind of see that all of her offspring were affected, okay? And so now it says, based on the pedigree, identify all the individuals in generation 4, so this generation right here, who can pass on the mutation to their children, okay? Now noticing that we have mitochondrial inheritance, that means that the mother is the only one can pass it on, okay? So if we look here, 3-2, can individual 1 be affected? Well, yeah, because the fact that this one is affected and she's going to pass it on to her offspring. So 1 is affected. If we look here at 2, 3, 4, individual 6 right here is also affected. So 2, 3, and 4 are all also affected, okay? Then we look here at 7 and 8. 8 is affected, but it's a male, so he can't pass it on. So these ones are going to be not affected. And then over here at 10 and 11, the parents are not affected, which means, of course, the kids are not affected, okay? So we need to figure out which individuals can pass on the trait. As we said, only females can pass it on. So that would be individuals 1, individual 2, and individual 4, okay? So that would be 4-1, 4-2, and 4-4. Because they asked about generation 4, you could have just written 1, 2, 4, like many people did in the chat. I mean, you would have been perfectly fine. So, based on the fact that individual 2 is affected, student claim that the disorder is actually X-linked. Okay, why is it not 3? It's not 3 because although he's affected, he can't pass it on as mitochondrial inheritance, which means only the female can pass it on to her offspring. Okay, so based on the student's claim, predict which of the individuals in generation 3 would be affected by the disorder. Okay, and then on the pedigree. Okay, so I'm ahead of myself. So here we're looking at right here, right? So if it was X-linked recessive, would this female be affected? Well, no, she's not going to be affected because of the fact that she needs to have 2 X links, 2 of those faulty X chromosomes, and her father is not affected. So she's not going to be affected. If we look at 4 is going to be affected because of the fact that he gets his X chromosome from his mother, and he only has 1 X chromosome. And the same thing with 6 that we saw with 2 is that she can pass on her X, but he doesn't have a faulty X to pass on. So 6 is not going to be affected. And then 8, of course, will be affected. Okay. So then we're going to see the 3-4 and 3-8 are going to be the 2 that are going to be affected if it was X-linked inheritance. And the reasoning being on this had to do with the fact that in order for 2 and 6 to be affected, the father had to be affected. So the data does not support the claim because the individuals 3-2 and 3-6 have the disorder because of X-linked inheritance. They would not have because the fact the father would have had the disorder, which he does not have it. Okay, so on to replication. Okay, so we are going to speed run this because I am already running behind. The location of this will take place wherever the DNA is. So in terms of eukaryotes, it's in the nucleus. In terms of prokaryotes, it's in the nucleoid. Prokaryotes are going to have a single circular DNA, versus a eukaryote is going to have multiple linear DNA. Students oftentimes screw this up. Okay, DNA is always red. Always red, 3 to 5. That 3' N is wherever you see that hydroxyl group, and the 5' N is wherever that phosphate is. Okay, you will always read 3 to 5. You cannot do anything else but read 3 to 5. Okay, now DNA is anti-parallel, which means that if you're reading it 3 to 5, then that means that we're going to synthesize it 5 to 3 because it's anti-parallel. They're going to be opposite, right? And so that's kind of what we're looking at here. Okay. And so there's a couple important enzymes. Single-stranded binding protein is not in the CED, as well as primase is not in the CED. In the CED, they have helicase. Helicase is going to unwind the DNA. It's going to break the hydrogen bonds between the 2 strands to pull it apart. Okay. Topoisomerase is going to relax the strand upstream. Okay. So that it's not going to get super coiled. Primase, as we said, is not in the curriculum, but what's going to happen is it's going to make our RNA primer. The reason why we read it 3 to 5 is because of the fact that the enzyme, DNA polymerase, that makes the DNA polymer. Remember, enzymes tell you what they do. DNA polymerase makes a DNA polymer. Okay, so DNA polymerase can't work unless it has an open 3' N to build off of, right? And so it has to work reading 3 to 5 because it can't build it in any other direction but 5 to 3. Okay, and then ligase is going to still again your Okazaki fragments, which will be on your lagging strand. Okay, so we have a leading strand and a lagging strand. When I say leading strand and lagging strand, all I'm talking about is that one strand is going to go into replication fork. Okay, so if this was my DNA, right? And helicase opens up a little bit, okay? We're going to make the DNA going toward that replication fork. So this one will be my leading strand because it goes into it, okay? But the opposite strand is going to go the opposite direction, and that would be my lagging strand. Once it opens up a little bit more, this one can keep on going down, while this one has to go backwards, okay? And it's very hard for me to model with my hands. I'm doing my best. Okazaki fragments are the little fragments that are made on that lagging strand. And so since I made all these little fragments, ligase has to come in and then seal them back together, okay? Now, both of these strands, this is a common mistake students make. Both of these strands are still reading 3 to 5. Both of these strands are still making 5 to 3. Do not tell me the leading strand is 5 to 3. They're both 5 to 3. You got this, y'all. You're penguins. Okay. Both of them are 5 to 3. Okay. So in terms of transcription, this is where we're going to read the DNA. We're going to synthesize an mRNA strand. Again, we're reading DNA 3 to 5. We're making RNA 5 to 3. DNA can only be read 3 to 5. Do not screw this up. I believe in you. You got this. Okay. Again, it'll take place wherever the DNA is. So we have RNA polymerase. Enzymes tell you what they do. RNA polymerase is going to make a RNA polymer. Okay. The promoter is where the RNA polymerase is going to bind to start. And then we have these different things called transcription factors. Transcription factors are going to bind to kind of cause the RNA polymerase to come bind so we can then be able to go on. Yes, you do want to be a penguin. Anyways, after we go through this process of making that mRNA, we're going to modify it in our eukaryotes, okay? So this is post-transcriptional modification. So we're going to add a 5' cap. So the 5' cap is going to signal where we're going to start on the mRNA in terms of our ribosome binding to it for our process of translation. And then it also allows for it to leave the nucleus, okay? We're going to have a poly A tail. Poly A tail is going to extend the length of the mRNA so they can stay around a lot longer. And then splicing is going to cut out those introns, and so that we'll be able to synthesize, we'll be able to get out the junk that's not there. That leads us into alternative splicing. Alternative splicing has to do with that we're going to cut out these different introns or exons, and so we can end up making multiple different mRNAs from the same DNA strand, okay? Translation has to do with we're now going to take that mRNA, we're going to translate it, we're going to make a polypeptide chain from it. We're going to, which one is copied? They're both copied. Both 3' and 5', both strands are going to be copied. One of them is copied continuously into the fork. One of them is done the other way. Introns are non-coding information. They're just junk that's in the DNA, okay? And so in terms of translation, we're going to start at the AUG, at the start codon. We're going to elongate. It's then going to go through and it's going to keep on adding the next amino acid in. You're going to have the same codon chart on your test, okay? And then it stops at the termination. So go UGA, UAA, and then UAG. And UGA, of course, is a football team, University of Georgia, or whatever, a tribe. Anyways, point mutations, this is going to be at a single nucleotide. So a silent mutation means that there's no change in our actual protein. It's going to be the exact same. Missense means that we go from one amino acid to another amino acid. Nonsense means we go from amino acid to a stop codon. And then frame shift means that we're going to insert or delete, and that can then cause you to get onto a different reading frame. And so instead of reading the cat ran too far, now it says Takaufa. That makes no sense. So what that means is that we've now cut out one of those nucleotides, which then causes it not to be able to read in the right grouping of 3 letters, okay? And so different other mutations that can happen, you can have insertion where we insert a huge chunk, we cut out a huge chunk with deletion, we duplicated certain sections, we also like cut off from one and popped it onto another one. Yes, methionine is always the start. And then we're also going to see we're not non-disjunction, so we can see that we're going to see that you can have an extra set of the chromosomes in polyploidy, or we just have an extra chromosome of a certain one, okay? So in terms of operons, we have 2 types of repressible operons and inducible operons. These are only found in our prokaryotes. So if our repressible operon is getting turned off because of the fact that that product is there, this is looking at negative feedback. That material that we're making is already there, and so it's going to bind to the operator it's going to bind to the repressor to turn the repressor on, so then the repressor is going to bind to the operator to turn the operon off, okay? And then the opposite happens within our inducible operon. In terms of our inducible operon, we're going to see that we're going to have that we have the material there, and it's going to come back, and it's going to cause us to synthesize the material to break down that material, okay? And so the lactose is going to come back, it's going to bind to the repressor, it's going to turn the repressor off, so then the operon gets turned on, okay? And then, of course, biotech, we're looking at gel electrophoresis. There's probably going to be some type of diagram like this. You put them in at the negative end. They get drawn toward the positive end because DNA is negatively charged because of the phosphate groups. And then you're going to see the larger fragments will be up here by the wells, and the shorter fragments will be down here at the end because it's going to be pulling them based on their size. It's kind of like pulling them in and out of these little like holes in the material. PCR is going to be polymerase chain reaction. It's going to make us multiple copies of that DNA in a very short amount of time. Transformation, you did this lab in which you transformed bacteria. You put DNA that was outside of it in terms of little plasmid, and you then put it into the bacteria. So now we got some new trait, okay? And then DNA sequencing, we can sequence that DNA strand, okay? So in terms of Unit 7, this is on natural selection, okay? So stabilizing selection means that we're going to pick for the middle. So they'll give you some type of explanation about something, and they're going to talk about how the intermediate was like favored. And because of the fact that the 2 intermediates were not favored, we kind of stabilize at that intermediate, okay? Directional selection, there's something that's moving in a direction. So like the peppered moths, due to the Industrial Revolution, there was a darkening of the trees. And so those that were too light were now more likely to be consumed, and those that were dark, that already had the mutation for dark, the pepper moths, they could blend into their surroundings and they were less likely to be consumed. So we directionally move toward the darker ones, okay? And then disruptive selection, there's something that's disturbing the intermediates, and we see the 2 extremes kind of coming up. So maybe it's like the seeds where like the medium beaked bird can't get enough food, and so now we have the small and the large beaked bird, and we kind of selected for those 2 opposites. What's going to happen when the exam is probably going to explain something to you, and you then have to apply that information, okay? So natural selection just really is looking at the fact that individuals that have a more favorable trait are more likely to survive. Because they're more likely to survive, they will leave more offspring, and because they leave more offspring, we see over time there's going to be an increase in that trait in the population. So mutations, this will give us new genetic variation, new genetic variation. It's the only way we get new ones, okay? We also have migration. Migration is going to bring genes from one population to another population. That's immigration, emigration, okay? Genetic drift is going to be non-selective. It is random what's happening with our genetic drift, okay? We have bottleneck effect and we have founder effect. Bottleneck effect means that this big group, and for some reason, we've drastically decreased the population, okay? And so that can cause some of our harmful alleles to become fixed. It could cause us to lose some of our genetic variation because maybe for some reason we don't have that trait that would have allowed us to survive something in the future, okay? Founder effect, we have a small group that gets separated and brought somewhere else, and then they can generate their own new population, okay? In both of these, whatever that new population is not representative of the original population, okay? And so genetic drift, in terms of this, a large population is going to help us to avoid problems when it comes into the genetic drift, okay? So evidence of evolution, you can look at fossil evidence, so the age of the rocks, you can look at the decaying of the isotopes as well as the geological area or geographical area, okay? Biogeography has to do with where these things are found. Biochemical, you're looking at DNA or protein. What is the better one to look? The better one to look at is DNA because you won't be able to see these silent mutations in a protein. But so between DNA and protein, DNA is more accurate, but between like proteins and like morphological structures, proteins are more accurate. Okay. And so you're comparing the number of differences in it. Morphological is looking at homologous structures. So similar structures due to common ancestry. So a bat wing and a cat arm, they have the similar structure because of the fact they have a common ancestor. That's why we were born with a tail. Yes, I don't think you knew this. The reason why we have a tailbone is because for a while there in development, we actually had a tail. It just went bye-bye by apoptosis, but you know, we did have one. And then ancestral derived traits means that it came from our ancestor. Okay, so just like what I saw with the tail, is that our ancestor had, and so we have that trait, okay? So it's some type of ancestral trait versus like a derived trait happened just on your lineage, okay? Now, somebody asked a second ago about convergent versus divergent, okay? Convergent evolution means that there's no common ancestor, like there's no recent common ancestor, and they were in a similar environment. Due to that similar environment, that caused them to have a similar structure. So a flying squirrel and a sugar glider, one's a marsupial, one's a placental, okay? They're not related to each other, but they look very similar because they have a similar environment, and that's led them to have a similar trait, okay? Divergent evolution shows our common ancestry because they had a common ancestor and they diverged from that common ancestor. Between DNA and proteins, DNA is more accurate because of the fact that you can see the silent mutations, okay? So if a silent mutation occurred, it's going to still make the exact same amino acid, so you wouldn't see it as a difference in the protein, but you would see it as a difference in the DNA. Okay, so common ancestry, that kind of leads us back into that they all have memory mounted organelles, there's linear chromosomes, and then the genes have introns, okay? You could have a phylogenetic tree. This is a tree is showing us 100,000 years. They can ask you questions about things that can be rotated. So like the European brown bear and the polar bear can be rotated because they share a branch point. They share that common ancestor. They ask you how long ago this happened between all the brown bears of their common ancestor. And you can say like right here, which is like, I don't know, 3.3, 3.4, 100,000 years ago. They can also look at cladograms. So this right here is a cladogram. When you look at it, you're looking for the smallest number of differences. Smallest differences, you'll then put them together. They do have a branch point, which means they can rotate. So it could have been brown and black bear. It doesn't matter. I just put those randomly down. And then looking here, we see that there's 7 and 8 differences with the polar bear, but there's 33, 34, and 40 with the panda. So it tells me that the polar bear is next, and then the panda is going to be my outgroup, or the one that is going to be the least related to all the other ones. Okay. So Hardy-Weinberg, okay, the 5 fingers of evolution. This has to do with that we want there to be an extremely large population size. We want there to be random mating. I forgot my wedding band, but do y'all see? I have a penguin on my nail. It's okay. Anyways, so we have random mating. We want them to, there not to be any type of sexual selection, no mutations, no gene flow, so no immigration, no emigration, and then no natural selection. If you have not looked at the 5 fingers of evolution from Paul Anderson, you probably want to check out that because it's honestly a great way to help you remember the 5 fingers, and it goes through the 5 that I just mentioned. So with this, that means that if the conditions are met, then there will be no evolution taking place. So there is an equation. This is the Hardy-Weinberg equation. P plus Q equals 1, where P is our dominant allele, Q is our recessive allele. The formula sheet just says allele 1 and allele 2, I think. And then P squared plus 2PQ plus Q squared equals 1. Okay. And so that's our other one. Okay. This is an approximation. Okay. You are approximating, okay, what these different characteristics are. Okay. And so when you do this, you want to always start with Q squared. You'll solve your Q squared. You'll take the square root of the Q squared, which gets you Q. You'll then say 1 minus Q, which then gives you P. Then we'll do squaring the P, which will give us P squared. And then we get a 2PQ, which will get our heterozygous, right? And so then you can fill in the chart and you can answer whatever question it is that they're needing, right? So, I wrote it up here. That's okay. So Now, if they tell you all of them, yeah, polyandry is boson bio. If they tell you there are 300 homozygous dominant, 200 heterozygous, 100 homozygous recessive, you need to count the alleles, okay? This is a more accurate way to determine it, okay? Because you will actually be counting the number of alleles you have. So take the number that are homozygous dominant and multiply by 2. Why? Because they have 2 dominant alleles. Then you're going to add in the heterozygous because they have 1 dominant allele, and that will then tell you how many dominant alleles there are. And then you divide by the total number of individuals times 2 because that's the total number of alleles in the population, and that will give your P. And then you can do the Q doing the exact same thing, looking at the homozygous recessive, and then P squared will be the ones you already know. Like I know there are 300 that are homozygous dominant over a total of 600 individuals, so 300 divided by 600 gave me 0.5 for my P squared, and that's how you would solve for those other ones. Okay, so as I said, you will use this method if you've been given the genotype numbers. Okay, so in terms of reproductive isolation, they'll probably give you some type of path, some type of scenario, and in that scenario, we're going to see that they're not going to reproduce. Why are they not going to reproduce? Because pre-zygote bearers. There's some type of behavioral reason, okay? Because of they have different mating signs or songs, mating dances. There's something different about their ritual, and because of that, then they're going to not be able to mate. Temporal has to do with a different time. They mate summer, fall, night, day, whatever. Okay. Geographic. They're in two different geographical areas. There's a barrier that separates them. Because of that barrier, they're not going to come in contact with each other. Habitat isolation or ecological isolation. Okay. There's no barrier between them. They just live in different areas. So like one's in a tree and one's on the ground. Like there's just different, right? Mechanical. This just has to do with the anatomically they're not going to work. And then the gametes just won't fuse. These are all pre-zygotic. We are not having sperm and egg coming together. They're all happening before the zygote is formed. Post-zygotic means it's after we form that zygote. So reduced hybrid viability. The hybrid is not viable. It's sick. It's bad. It's going to die. Reduced hybrid fertility. It's like a mule. It's not going to be able to continue reproducing because it is not fertile. It is sterile. Okay. And then hybrid breakdown has to do with that. The first generation is perfectly fine, but it's going to break down. It's not going to be as healthy over time. Okay. All of this is decreasing our gene flow. Okay. It's making it so that they're no longer mating with each other. And as over time, they stop mating with each other, as there's a decrease in that gene flow, there can be mutations happening in one subpopulation that doesn't happen in the other, and that leads into speciation. If they're in the same area, it's sympatric speciation. If they're different areas, it's allopatric speciation. Okay. So then looking into unit 8, okay, we're looking at different types of communication. So we have visual communication, auditory, tactile, electrical, chemical. These are just different ways that they can communicate with each other. Okay. Why we need to show our dominance. We need to find food. We need to get a mate, right? We got to do a blue-footed booty dance in order to get a mate, right? Anyways, you also establish their territory, okay? Sometimes they're just responding to their environment with some type of behavior or psychological mechanism, right? So intersexual selection has to do with the fact that they're going to be choosy, okay? So in the blue-footed boobies, they're doing a beautiful dance. They're showing off the blue feet. That is behavioral isolation. If you have red feet, I ain't going to mate with you, okay? So they're going to choose the one that has the best feet because the best blue feet means that they have the best genes, and I want to mate with the one that has the best genes, right? Totally, okay? Anyway, so intersexual is one choosing the other, right? Intrasexual, yeah, I know you had to have another jump scare, didn't you? Intrasexual has to do with that we're fighting, right? So there's going to be 2 of them that are fighting for the ability to mate with the other, right? And so here you can see these 2 are fighting. The one that wins is going to be able to, of course, get to mate with all of the other individuals in that little grouping. And here you can see the little ants are fighting. So biogeochemical cycles was added into the curriculum. You do need to make sure you know these cycles, okay? So the water cycle is just looking how water goes around, looking at evaporation, condensation, precipitation, and transpiration. Carbon cycle, looking at photosynthesis and cellular respiration and decomposition and combustion, where we're going to, burn things. Nitrogen cycle, this is where we saw the nitrogen fixation, where we're going to fixate the nitrogen from the air into ammonia, and then we're going to make it into ammonium. Okay, nitrification, we're then going to make the nitrate, and then assimilation, we're going to get the nitrate to then go in, make our organic compounds, right? Ammonification then converts the nitrogen into ammonia, and then denitrification converts the nitrate back into nitrogen. Whew, okay. And then the phosphorus, we're just going to weather rocks so that we can get the phosphate out of it. The phosphate is going to be put into like different organic molecules. So thinking about like your nucleic acids or your phospholipids. That's where we're going to see the phosphorus at. And so you can kind of see it cycling through that process. Okay. So ecology has a couple of different calculations. Exponential growth. Exponential growth is going to be where it's going to kind of have unlimited growth. There's nothing that's going to limit the population. So that would be our R times our N, okay, where we're going to see our rate of increase, which is our birth rate minus our death rate, times our population size, and that will increase. Versus if there is a carrying capacity or something that's going to kind of slow it down, it's going to stop it from kind of going through that process. Like we don't have enough resources, we don't have enough nutrients, there's not enough space. There's something that limits that population. That's our carrying capacity. And so it's going to kind of level out over time, and that's what that K is. That K is carrying capacity. So we'll do our rate of increase times our population size, carrying capacity minus our population size over carrying capacity. Why is there a 200 there? I think that's because this is left over from when I forgot to delete it. Ignore this 200 right here. This should not be here. Ignore that. Please do not think it's always 200. This was just, I was pulling the PowerPoint I did from the last live basically yesterday. And then Simpson's index, we're going to looking at biodiversity. So we're going to see that we've got the summation. So we're going to add together the individuals that are in that species over the total that we have in the population. I'm sorry, in the community. And then we're going to square each of those and add them all together. And then students oftentimes forget to do 1 minus. Okay. So community ecology, this is where we're going to see the interactions. Oftentimes they'll give you some scenario. And this is when I find out things about animals that I didn't know. Like, did you know that the cuckoo bird just leaves its offspring in a warbler nest for the warbler to take care of? Yeah, I didn't know that either until I read the AP exam. Cha-ching, learn biology. Anyways, so we have predator-prey reaction where the predator is going to eat the prey. Bye-bye prey. Herbivory where the herbivore eats the plants. So bye-bye plants. Competition, they're both competing over some type of resource. So we're going to see negative impacts of both of those. Parasitism, the parasite is going to take resources from the host. And so what's going to see is that we'll see a decrease in the host and increase in the parasite. And when I say increase, I mean like a positive influence. Mutualism, they're both benefiting each other. And then commensalism, one of them benefits, one of them has no effect. Okay. And so they're going to kind of interact together so they can access appropriate nutrients as well as they can work together within their community. They want to cooperate. So they have some type of common goal. So mutualism, they're working together, like the digger crab and the sea urchin. The sea urchin will hang on the back of the crab. The crab will give it movement so it can move somewhere else, and the sea urchin provides protection for the crab. Commensalism, they're going to kind of benefit from each other. Altruism, where one of them is going to have some type of selfless behavior that then helps the others. Okay. Top-down model, we're going to see that our predators are going to control everything. In the bottom-up model, means our producers are going to control everything. And then niche partitioning, you can't have two individuals or two different species working on the same thing. Working on the same area, they're not going to be able to occupy the same niche, so we have to break up that niche in some way. Okay, so we got the practice problems, and then we were done. Okay, hold it with me. So humans have 23 pairs of chromosomes, which following statements best predicts the consequence if correct chromosome separation did not occur during meiosis 2. Okay, so here's the thing: they have 23 pairs of chromosomes, right? If I don't appropriately separate them, what's going to happen? Well, let's see your answer choices. I don't know. The gametes would get larger from one generation to the next. Well, the gamete isn't going to get bigger because this is just looking at chromosomes. Don't separate. Okay, chromosome number will double each generation. So if at the end of meiosis my cell was diploid and I had two diploid cells coming together, now the organism is going to be tetraploid, and then the next generation is going to be, I don't know what 8 would be, octoploid. And so like it just keeps on building. So B sounds logical. Let's look at C. The chromosome number would be half each generation. Well, it wouldn't be halved because I didn't decrease it because the separation didn't happen, and then it wouldn't triple because I'm going to have an extra one here and an extra one there. So it'd actually be to the 4, like it would square it each time. So B is our only logical answer. Okay. So then mutations in these two different genes. Again, do you really need to know what they are? No, it's X and Y. These genes have associated with a form of hearing loss. They say the genes that are proposed enclose a transcription factor that regulates the myo6. Okay. So we know that this one, the P, is going to make a transcription factor that then regulates myo6, which the following best helps me guide the researchers toward a direct test for their proposal. Okay, again, we're going back to the transcription factor. Have mutations in other genes been associated with hearing loss? I'm trying to figure out this transcription factor thing, so don't care about other mutations. In what types of cells are the mutant forms of the gene expressed? Again, I want to know about the transcription factor. Are the mutations are they both in the mice? Well, I hope so. Do the mutations in the gene affect the mRNA levels? Well, if it's a transcription factor, that means that it's going to be binding to allow the RNA polymerase to bind so I can then turn on transcription. So if a mutation in it affects the mRNA level, that's going to tell me whether it's a good, if it's going to work as a transcription factor. So D is my answer on this one. DNA replicate during the S phase, but it's still considered diploid. Yeah. So human TPM gene encodes members of this tryptomyosin family of our proteins, which follow best explains how the different proteins can be made in different cell types from one gene. Okay, so I have one gene, but I'm making multiple different types of protein from it. So different introns are selectively converted into exons. Maybe. Different exons are retained or spliced out of the primary transcript. Okay, whoa, that's almost splicing. That makes me think of alternative splicing. So there's different exons, and because of what gets kept and what is cut out, then we're going to see differences. I like B so far. Let's keep going. The GTP cap is selectively added to and activates different exons. No, the GTP cap is just added so that we know where the ribosome is going to bind. So no, I don't like C. Different portions of the primary transcript remain bound to the template DNA. What? No, that's not helping. I like B, y'all. Let's see. B's right. Yes, we're smart. I'm so proud of y'all. Okay, so different photosynthetic organisms have different types of chlorophyll molecules. The distribution of chlorophyll in several different groups of organisms is shown in the table. A plus sign on the table indicates the presence of a chlorophyll, while a minus sign indicates its absence. Okay, so which of the following most likely describes the evolutionary relationship among the organisms? Okay, so we're looking for evolutionary relationship. If I look here, I can see that they all have chlorophyll A. So that must mean there's some type of common ancestor. And due to the common ancestor, we're going to have that they all have chlorophyll A. We can see that the flowering plants and the green algae are related to each other, and the brown algae and the red algae are probably also related. We see that this brown algae somehow has chlorophyll C, and the red algae has chlorophyll D. Those were probably just different types of traits that may have happened on their lineage and only their lineage, so there's some type of derived trait. Because brown algae, red algae, and cyanobacteria lack chlorophyll B, they evolved before the green algae and the flowering plants did. There's no evidence that proves that they evolved before it. They may have diverged and one branch got it, but the other one didn't. Because green algae and flowering plants contain chloroplasts, they evolved more recently than brown algae, red algae, and cyanobacteria did. No, I don't really know. We can't say that. Because increasing complexity forms of chlorophyll are found in red algae, brown algae, green algae, and flowering plants, evolutionary relationship among these can be seen over time. We still don't know if that's actually happening. Because all the organisms contain chlorophyll A, the organisms, hey, I said that! D's our answer. Which person is lying? Okay, so apple maggot-fly, okay, it's native to our North America and originally fed on the fruit of a wild hawthorn. Since the mid-1800s, the population of flies emerged and they feed on domesticated apples. Okay, so there's some group of them where the apple maggot-flies mate on or near the fruit of their host. Many varieties of apples ripen several weeks before the hawthorn fruits do. Different fruits preferences of the two flies will have a common effect. Okay, so what you see is we have a fly. We have an apple maggot fly, and then we also have a hawthorn fly, and they're going to be on different fruit. And so since they're on different fruit, are they going to mate together? Okay, cool. The flies that eat hawthorn fruit will increase in number, while the flies that eat apples will decrease in number because of the use of insecticides. We said nothing about insecticides. No. The single fly species will evolve into two distinct species because of the lack of gene flow between the two populations. There's a lack of gene flow. They're going to have habitat isolation. They're on two different habitats, and so since they're on different habitats, they're not going to mate with each other. I like B. The ability to survive on the diet of two different fruits help them learn to eat more fruit. No. The flies that eat hawthorn will lay some of the eggs that are earlier ripening to maximize competition. Again, no. They're only competing against themselves. So B is our answer. Okay, so this is our set question. Okay, so it gives us this little background. We've got this like graph over here, or this data, like what is this, a diagram? And it talks us about how carbon was a limiting nutrient in an aquatic ecosystem. To test hypothesis, researchers divided a lake into two different equal halves. With a curtain put in between them. They then treated one side with sucrose and the other with sucrose and phosphate. And they monitored the phytoplankton biomass in both parts, and then the results were shown right here. Okay, so which of the following claims is best supported by the data? Okay, so looking at the data, what is supported by this? Okay, so I see that when I'm treated with sucrose, it's low. But if I have sucrose and phosphate, it's high. So there's something about the phosphate that's allowing it to grow. Okay, let's see. Carbon is a limiting factor for the phytoplankton in the lake. There's no evidence that shows that. Phosphate was a limiting factor. Well, I need phosphate. And if I don't have any phosphate, then it's not going to grow. Okay, cool. Both carbon and phosphate were limiting factors. No. Neither carbon nor phosphate was a limiting factor. Okay, again, we need phosphate. If I have the phosphate, I'm high. If I don't have the phosphate, I'm low. So phosphate must have been a limiting factor. Oh, I got somebody on the D. Okay, so average growth rate. Okay, again, Ms. Jones, why do you keep giving us things? Well, the rate formula is on the formula sheet, okay? Like, just pay bear with me, okay? So we need to figure out from 1971 to 1975, and we got to get this information from the data, right? So if you look here at 1971, right? I didn't draw the down arrow, but here's 1971, right? It looks like we have about 1,000, right? So then if we look at 1975, we have about, what's that, 7,000? So then it's 1975 and 7,000. So what we're going to do is we're going to do the change in Y over the change in X, okay? And so with this, we're going to see that we're going to do our 7,000 minus the 1,000, so change in Y over our change in X, 1975 minus 1971. That gives us 6,000 over 4, which gives us 1,500, so the answer is C. Okay, so then in the early 1970s, they hypothesized that the carbon was a limiting nutrient in our aquatic ecosystems. To test the hypothesis, they divide a small lake into 2 roughly equal parts with a curtain to fasten it. The researchers treated one side of the lake with the sucrose and the other side with the sucrose and the phosphate, and then they monitor it. So I just basically took that prompt and gave it back to you. Which volunteering would be the best control? Okay, so what I did is I highlighted what they did, right? This was our independent variable. We had our sucrose and the phosphate, okay? So I need to find a control for this, right? So how do I know what happens normally? I know what happens with sucrose. I know what happens with sucrose and phosphate, but I don't know what happens when they're not there. So untreated section of the lake, section of the lake that's treated with phosphate but not sucrose, a different lake that was treated with sucrose and phosphate, again, I already have that, a small pool of the lake maintained in a control environment. If it's maintained in a control environment, it's not following the other materials that happen. So I like A. A is our answer, and you are all rocking it with all those A's. I love it, okay? Which of the following is most likely a direct consequence of the addition of phosphate to the lake, okay? So why do I see, if I add phosphate, what's happening, okay? Well, I see that it's increasing, so the amount of biomass in the first trophic level decrease. Well, phytoplankton, that would be on my first trophic level. Is it decreasing? Well, no. I see an increase in my biomass. There's way more of them. The amount of biomass in the second trophic level decrease. Well, if I don't have enough food, it will decrease, but I should have enough food. The amount of energy available to the producers in the lake increased. Well, if I have more biomass, that means there's more stuff for things to eat. Biomass is like the amount of the phytoplankton that we have. Like it's the overall, all the phytoplankton is like how much of there it is, right? It's all the energy, right? Or the amount of energy available to consumers in the lake increased, okay? So we're looking at the amount of energy that's available to the producers or the amount of energy that's available to the consumers. Well, this right here is my producer, and so is going to be consumed by my consumer, so it should be D. Anyway, I got a lot of y'all on that one. And so that is the end. I'm so sorry I went over by 8 minutes, but I think I did pretty good getting everything done in 2 hours. So if you need to go through any of the other sections, you need to go and, re-watch any of my long form videos. They are right here in this QR code. You can scan that. It will also bring you to where this recording is. It will also, gives you the PowerPoint in case you're going back to it. If you want, again, those quick reviews, there's a TikTok. Good luck on the exam. Y'all are going to rock this. Your penguins, you're dressed for success. I believe in every single one of you. I hope that you come back and tell me how you did, because I know we're going to get so many 5s, because I love you all, and I know that you are going to rock this thing. You're going to do your best. You're going to leave it all on the test, and you're going to remember that you're AP Bio Penguins and you're dressed for success. So I hope that this was helpful, and I will see y'all later. Bye.